On behalf of the Department of Microbiology and Medical Zoology of the University of Puerto Rico School of Medicine, it's a great pleasure to greet you all to the Microbiomes from the Environment to Human Diseases mini-symposium, sponsored both by the UPR RISE graduate program sponsorship from the NIH to fund graduate students, and the AAAS Caribbean Division, and also Vistazo a la Ciencia. It's a pleasure for me to greet you all this morning, and thank you for taking the time to learn from the best on microbiome research, a new and exciting field of microbiology. Buenos días a todos, muy bienvenidos a nuestro simposio de microbioma, organizado por RISE y por el Departamento de Microbiología. Para nosotros es un placer. Somos el único departamento de ciencia en el Caribe que ofrece proyectos y entrenamiento a estudiantes en el field de microbioma, que es un área nueva de la microbiología reciente, un área no tan nueva, en los últimos 15 años más o menos apareció este nuevo field de la microbiología y estamos muy contentos de traerles el primer mini-simposio en Puerto Rico sobre este tema. Traímos los expertos mundiales y los que desarrollaron paquetes de análisis de datos que son usados universalmente para el estudio de microbioma. Así que espero que disfruten y que nos acompañen durante la mañana de hoy hasta el mediodía. Después, por la tarde, de 1 a 2, va a haber un lunch break, y por la tarde hay una sesión más para estudiantes donde vamos a hablar de career development, oportunidades en el CDC, oportunidades en la academia, en las mismas farmacéuticas. Aquí en Puerto Rico vamos a tener personas con diferentes backgrounds y que están haciendo cosas muy diferentes para darle un poquito el estímulo a nuestros estudiantes graduados y que vean que la microbiología no es solamente o la academia o la farmacéutica. Hay una gran diversidad de carreras que se pueden seguir, y especialmente los que saben de datos, de análisis de datos, tienen las puertas abiertas a un mundo nuevo de trabajos muy bien remunerados también. Así que queremos hablar un poco de eso y vamos a tener nuestros estudiantes graduados participando activamente como moderadores de las sesiones. Yo soy la primera a moderar esta primera sesión de la mañana como directora del Departamento de Microbiología, y me place llamar, no sé si está ahí el doctor Guillot, porque no puedo ver la pantalla. Buenos días, doctor Guillot, nuestro decano. Le quisiera pasar la palabra para que nos dé la bienvenida protocolaria y nos dé unos minutitos, unas palabras.
Sí, hola.
Hola, buenos días, ¿qué tal? Muchas gracias por venir.
Okay.
Yo puedo decir, antes que empiece, solamente quisiera decir a la audiencia, I would like to tell everyone that you're also a member of our department. You're a professor at the Department of Microbiology. You're an expert in anal cancer. And I have the privilege of collaborating with you in the Campo, an NCI-sponsored clinical trial. It's going to be the first clinical trial in Puerto Rico on human microbiome. So I'm very excited. I'm very thankful that you're here today.
Okay. Thank you very much for the invitation, and welcome you all to this mini-symposium organized by the RISE program and the Department of Microbiology and Medical Zoology from the University of Puerto Rico School of Medicine. We are delighted to have you all in this virtual experience from the environment to the human disease. During today's session, you will be discussing all the diversity of this novel yet promising and developing topic, microbiome. I'm an infectious disease specialist, like Felipa just said, and I'm a co-joint professor in the Department of Microbiology. And this was certainly not a topic when I was doing med school or even my fellowship in infectious diseases. So I am confident this will be a very enriching opportunity for all. And thanks again for joining us, and enjoy your sessions. Bienvenidos todos. Bienvenidos todos a este simposio organizado por el programa RISE y por el Departamento de Microbiología y Zoología Médica de la Escuela de Medicina de la Universidad de Puerto Rico. Estamos bien orgullosos de ser parte de este simposio, de que la doctora Filipa Godoy, jefa del departamento, lo haya organizado junto con sus colaboradores, que se llama Del ambiente a enfermedad humana. Vi el programa durante las sesiones de hoy, van a estar discutiendo toda la diversidad de este tópico tan novel y a la vez tan prometedor y que se sigue en desarrollo, que es el microbioma. Yo soy un especialista en enfermedades infecciosas de adultos, soy profesor conjunto del Departamento de Microbiología y ciertamente el microbioma no era un tópico cuando yo estudiaba medicina y no era un tópico tampoco cuando estaba haciendo mi fellowship en enfermedades infecciosas, pero ciertamente hoy es un campo en desarrollo que promete brindar tantas explicaciones a enfermedad humana y también a terapia. Estoy bien confiado de que la sesión de hoy va a ser una bien enriquecedora para todos los participantes y nuevamente les agradezco por unirse y agradezco a todos los organizadores y a todos los participantes que disfruten muchísimo todas sus sesiones. Así que gracias.
Muchísimas gracias, doctor Guiot. Gracias. Para nosotros es un privilegio que nos diera este mensaje, siendo usted un especialista en enfermedades infecciosas y nuestro decano de medicina. Muchísimas gracias. Que tenga un buen día, doctor. Nuestro próximo speaker es la doctora Nivia Pérez Acevedo. Ella es la decana de Ciencias Biomédicas y directora del programa graduado. Nos ha llevado de la mano en la organización también de este simposio y nos gustaría que ella nos diera también unas palabras esta mañana. Doctora Pérez, ¿está por ahí? Yo no veo la pantalla. Alguien me tiene que ayudar aquí.
Está por ahí.
Aquí ahora, ¿me escuchan?
Hi, good morning.
Muy buenos días a todos y todas. Voy a cambiar a inglés. For the general public, microbes are almost always, I think, a bad thing, right? In pandemic times, due to widespread misinformation, some people may think that COVID-19 is in fact a microbe. Other people in our communities may have been taking the recommendations made by the U.S. Center of Disease Control and Prevention or by our local public health specialists to the point that sequestering themselves into a hypoallergenic home. Why? Because they are afraid to microbes. It is the duty of biomedical scientists to tease apart the science and the fictions of microbiomes from the environment to human diseases. The Associate Deanship of Biomedical Sciences of the School of Medicine of the University of Puerto Rico is most grateful to Dr. Caporasso from Northern Arizona University, to Dr. Kinney from the University of Texas at Austin, to Dr. Landay from Rush University and Medical College, and to Dr. Crandall from the George Washington University for their time and ongoing conversations with our scientists in Puerto Rico. Due to unprecedented health concerns in this 21st century, this symposium is particularly timely. Due to these same health concerns, this important meeting is an online live event. But I truly hope that this will be the first of many scientific exchanges so that we can properly welcome you to Puerto Rico, to our institution, and to the biomedical faculty and students who work from these latitudes of the world. Given the roster of national and local experts, I already celebrate this meeting and I look forward to the outcomes in benefit of our scientific and general public communities. Thank you very much and enjoy this symposium.
Thank you very much, Dr. Perez. All you have said is so true and is so exciting. I agree with you that it's a unique opportunity to extend branches with these experts that could, help our students and help with their scientific development. And we truly hope this is just the beginning of new collaborations and many other exciting venues. Thank you so much.
Thank you. Have a great day.
Muchas gracias.
Gracias a todos.
Ahora le quería dar la palabra a la doctora Carmen Cadilla. Ella es nuestra host en el día de hoy. Ella es la PI del programa RISE. del NIDCR. Ella es bioquímica también, científica, y es nuestra líder, fue también decana de ciencias biomédicas y está ahora dedicada a la ciencia, y me gustaría que nos diera también unas palabras esta mañana. Muchas gracias, doctora Cadilla.
Buenos días y gracias por la invitación. Voy a cambiar ahora a inglés. I would like to welcome you to the third NIGMS RISE program mini-symposium, which is hosted by the Department of Microbiology and Medical Zoology of the University of Puerto Rico School of Medicine. As Dr. Godoy said, I am the RISE program director and also professor of biochemistry, and I'm also the RCMI program genomics service lead for the Medical Sciences campus. This is our first online mini-symposium, as has been said, due to the conditions that we are presently. Focus will be on microbiomes, from the environment to human diseases. The UPR Medical Sciences Campus RISE program is currently in its 21st year of continuous NIH funding and has provided support to hundreds of underrepresented minority PhD students. The vast majority of them are presently engaged in research careers in academia, government agencies, and industry. One of our main goals is to contribute to bring our students and faculty visiting speakers like the ones we're having today, who are involved in cutting-edge science. The graduate students of the Division of Biomedical Sciences contribute greatly to the scholarly activity of the School of Medicine and the Medical Sciences campus. The microbiome refers to the collective genomes of genetic material of all microbes in a particular environment called the microbiota. Since we are located in a medical school, we frequently focus on the human microbiome, which describes the collected microbiomes of the human body that reside primarily in the gut and that vary considerably from one individual to another and among different anatomical sites. Factors that are known to influence the microbiome include diet, lifestyle, genetics, anatomical site, disease state, antibiotics, and pathogens, among others. Today's mini-symposium distinguished invited speakers will cover different aspects of microbiomes, including the gut microbiota and its viruses, HIV immunopathogenesis and aging, microbial exposures and occupant health, and computational approaches for characterizing microbiomes. We are very grateful to the faculty and students of the Department of Microbiology and Medical Zoology for organizing this exciting mini-symposium, and to the distinguished invited speakers and special guests who hail from places like Arizona, Texas, Illinois, and our nation's capital, who are honoring us with their participation, and all who are attending this event. We welcome to our island, although virtually. Hopefully in the future you will be able to visit us presentially. And also the medical side, visiting us, we're also visiting the Medical Sciences campus of the University of Puerto Rico, apart from our island. So welcome to all, and I hope this event is as successful as it's going to be. Thank you.
Thank you so much, Dr. Cadilla. I have a short meeting introduction on the microbiome, very, very short, because I was expecting naturally some delays. I'm just emailing the first speaker, who is not yet in the line for in Round the World. So give me just 1 second.
I believe he just arrived.
I'm here. It's
oh, okay.
Is present.
Issues getting on.
Welcome, Keith. Good morning. How are you?
Good morning.
Thank
Good,
you so
good.
much. And thank you all our amazing speakers this morning. Dr. Keith Crandall, Dr. Alan Landay, Dr. Kerry Kinney, and Dr. Greg Caporasso. First, I'm going to just give a little introduction to what is the microbiome, and also to our microbiology department. This is a unique opportunity to showcase our department for prospective students and for those who are joining us from everywhere around the island and outside Puerto Rico. I'll have to thank you again, all the leaders that have joined us this morning, Dr. Cadilla, Dr. Perez, Dr. Guillot, for their great incentives and great support in developing the symposium, and for taking the time to greet us this morning. I'd also like to, of course, to thank RISE, our main sponsor and organizer of this event. RISE sponsors graduate students. It's a program from the NIH, for those who don't know, for minority institutions, and his basically the support for many of our labs. So thank you very much. I'd like to thank especially Barbara Santana and Dr. Jorge for their amazing leadership and for bringing us hand by hand every day in making this happen. I would also like to thank the AAAS Caribbean Division for helping us sponsor the event and also share the agenda and the plans of our meeting with everyone across the social networks. And also Vistazo a la Ciencia and his leader, Dr. Felix Rivera Mariani, who is helping us produce this event today and has been a great partner in this last months. So thank you all so much. So this is the Department of Microbiology and Medical Zoology. It's on behalf of this department that we do this event today. It's part of the University of Puerto Rico School of Medicine. And as I said in the beginning, it's the only program in the Caribbean that offers training in microbiome, in human microbiome, and in environmental microbiomes as well. We are truly honored that you are joining us this morning, and we would like just to do a short overview of what the department is. It aims to train the next generation of microbiologists in the frontier of basic and translational disciplines. It emphasizes a bridge between students and faculty. We want to expose students to being leaders and to give them opportunities to excel and to shine. And therefore, we give them opportunities to go abroad, to get trained, to participate in these types of meetings. In fact, our students are being moderators in all these sessions. We have a creative vision that we want to see evolve in order to recognize all this talent and this amazing talent from all these students from different backgrounds, with different training experiences, in order to empower them to be the next leaders in the scientific and technology fields. This is a summary of our professors. We are a small department, but nonetheless, we are productive and we are very supportive of student development. Our department has 10 professors, ranging from parasitology, mycology, bacteriology, virology, immunology, cancer immunology, and microbial ecology. So if you are a prospective student, I urge you to go to our website or to email us directly for information. We will be glad to share your concerns and answer your questions. Our graduate students are involved in many activities in leadership, in the community, doing a lot of outreach activities, conferences, networking, and basically they help other students. It's a lot of active support between senior to more junior students. They help each other a lot, and they do a lot of activities, and they also try to make the environment a nice and exciting one to work. We do a lot of oral presentations in local and national international forums, and publish in peer-reviewed journals. So it's like working in any U.S. institution. The bright side is that we have sunny weather, and we have tropical temperatures all year round. So if you're out there, if you're thinking about joining a microbiology program, come to the Caribbean. So microbes, we're doing this to celebrate the amazing microbial diversity that is now assumed to be part of all living things. So microbes predate Earth's biodiversity. This is the slide that I always show in my talks. Microbes are the oldest organisms on Earth. They were the first here, about 3.7 billion years. And everything that evolved after the appearance of the first prokaryotic cells have evolved in the presence of them. So all the multicellular eukaryotes, animals, plants, and us humans, we are colonized by a plethora of different virus, fungi, bacteria, archaea that make up a lot of our functions and make a lot of the functions that we as vertebrates don't do. So microbiology touches everything. The microbes are in us and on us. They are everywhere, and they do a lot of the functions that we cannot do. They have enzymes that are capable, for example, in cows and in forget fermenter birds, they have enzymes that break down cellulose in order for these animals to produce energy. Like even in the termites, there's a very complex microbial diversity in their guts to digest the wood. In the sea, in our guts, they're the most famous microbes. It's in the human gut microbiome, but we also have other body niches. As said previously, we have different body niches depending on the physiology of our bodies, and specific populations trigger functions that we need in order to survive. So then comes the microbiome, which is a new field about 15 years ago. After all these sequencing machines were resting after sequencing the human genome, and then an international group had the idea to start sequencing microbes. So a new field was born and opened our eyes to the amazing diversity that underlies many, many human diseases, but also our states of health. So the collection of all these bacterial cells, it's called the microbiota, especially when we use gene sequencing, specific genes, biomarkers like 16S, or the hypervariable regions of the 16S, or when we sequence ITS or 18S, we are just using one gene.
about some of our work in the microbiome space. As Filipa was introducing the concept of microbiome, it's really been important in a wide variety of areas, and I'm going to touch on a few of these different areas, but also on computational methods. I'm Keith Crandall, and I direct the Computational Biology Institute at George Washington University, and I'm also a professor in the Department of Biostatistics and Bioinformatics in our School of Public Health. And I have my lab does a variety of things, including method development, but then applying these methods to a variety of different study systems, some in human health, cancer, infectious disease, but also natural history studies and natural history collaborations. And as a consequence, I'm also a research associate at the Smithsonian in the Department of Invertebrate Zoology. So I'll try and give you a diversity of these different applications, as well as an introduction to some of our methods. Filipa was just telling you about why we're interested in the microbiome, that it's this incredibly important aspect of human health, and even weird things like human behavior and organismal behavior. People have been interested in the microbiome in terms of organismal microbiomes and also environmental microbiomes. In humans, a variety of diseases seem to be associated with microbiome diversity. And we're specifically interested in, given this importance of the microbiome in a variety of areas in biology, how do we go about surveying the microbiome? And we also specifically focused on microbiome and cancer, and published this review paper a couple of years ago now. And this is a nice example of how the microbiome can impact human health, but in a variety of different ways, as a causal factor, as an impactor in terms of treatments, and in terms of diagnosis. So it's really an interesting area to study, very exciting area to study. And so we're excited to tell you about some of the methods that we work on. So a typical kind of study that we look at in our group is this particular study is a partnership with Children's National in Washington, D.C., where you have some kids who have asthma. And of course, now in the SARS era, we're all familiar with with getting swabs stuck down our nose for COVID testing. But of course, this is how we sample for microbiome as well. Take that swab, swab some noses, sequence that snot, and get millions and millions of short DNA sequence reads. These are usually about 150 base pairs. And then what we try and do is figure out what's in there, what's in that snot, by virtue of surveying these DNA sequencing reads. So this is shotgun metagenomics, as opposed to the targeted amplicon sequencing, where you're actually extracting DNA from the nasal swab and then doing a PCR to sequence one particular gene. Here we're doing shotgun sequencing. So we just sequence everything that's in that nasal swab. And then the trick is, what do we do with all these sequencing reads? So originally people started trying to assemble these things because computational biologists dealing with next-gen sequencing were all about genomes and genome assembly, and most of the software, frankly, is targeted towards doing assembly. But assembly is hard, especially when you have low coverage short reads. So we thought, let's not assemble those genomes. Let's just take those raw sequencing reads and map them to a reference library and figure out which reference genomes these sequencing reads map to, and then we can identify which microbes are in this population. And the nice part about this shotgun sequencing is you can survey everything that's in this kid's nose, right? You can survey fungus, virus, bacteria, everything that's there by doing the shotgun sequencing, as opposed to the If you want to do targeted amplicon sequencing, if you want to look at bacteria, you do a PCR reaction on 16S. If you want to do fungus, you have to do a PCR reaction on an ITS region. So there are different gene regions that we target for different organismal groups. But with shotgun sequencing, we just get everything at once. And so you can look at ATCC has some prepared diversity microbial communities, right? That you can look at, and then in here it's 10 organisms of even ratios, and then you can do the amplicon sequencing of 16S. These are all bacteria, and then the shotgun sequencing, and you can show that shotgun sequencing actually gets you back to what the actual composition is better than the 16S. So the shotgun, not only does it allow you to survey more with a single effort, but it gets you a better estimate of what's actually in the original mix. And that's whether you do 10 organisms or 20 organisms, and you get similar results with the ITS, although the ITS isn't quite as pronounced in terms of both the shotgun and the ITS have issues. And you can also explore the same concept across different microbiomes: oral microbiome, skin microbiome, gut microbiome, vaginal microbiome. So when we talk about the human microbiome, you have to be specific, because of course you have a diverse microbiome depending on which part of the human you are actually sampling. And again, the shotgun methods really get you better, closer to the reality of a predetermined mix than the 16S. So we try and convince folks to do shotgun sequencing. The difficulty, of course, is the pricing. The 16S sequencing is super cheap compared to shotgun sequencing. But given these kinds of data, these short read sequences from either 16S or shotgun data, we've developed some software called Pathoscope that allows you to do species level and even strain level identification by doing some sophisticated mapping of these sequences. So you can think about this the naive way. In fact, our baseline comparison is to just blast all your sequences against a reference database and see what comes up. And that's, a lot of people do that, and we're doing that originally, and that does not perform well, at least not relative to our approach. And so we developed some software called Pathoscope, which is freely available on GitHub. And the meat of Pathoscope is this Bayesian mixture model. And the real crux of our approach is this reassignment parameter that allows you to take reads and reads that map to multiple genomes in your reference library, non-unique reads, and reassign them to genomes where unique reads are mapping. So the biggest issue with these kinds of approaches is you have unique reads and non-unique reads. And most methods either do one of two things. They either throw out the non-unique reads because they're not as informative as the unique reads, in which case you're throwing out most of your data, or they include the non-unique reads and have much reduced resolution because the non-unique reads map all over the place. So they end up having a lot of false positives with those methods. So our approach does this reassignment. And the basic idea is if you have non-unique reads, these red ones that are in mapping to 3 different genomes, but you have some reads that are mapping to just 2 genomes and some reads that are mapping to uniquely to a single genome, then all those non-unique reads get assigned to that unique genome. And then you can do the same thing if you have unique reads in 2 different genomes, then you just split the non-unique reads in the frequency of the unique reads across those 2 genomes. So that's the concept. And we put this together in software called Pathoscope that starts with these raw sequence reads that you have. It builds a library, a reference library. We usually use NCBI, although you can use any reference library you want. You can build your own custom library. You can use RefSeq from NCBI, or you can add additional sequence data from NCBI. But the reference library tends to be fairly critical. Like all good bioinformaticians, we don't reinvent the wheel. We went with a mapper that's already available, Bowtie 2, and that seems to do reasonably well. But we apply our Bayesian mixture model after that initial mapping, and remap some of the reads based on those posterior probabilities. And then we get some assessment of the diversity in the sample. So we've tested our approach against other approaches using data from the human oral microbiome database. So we took the raw sequence reads from these and the calls, and we fed the raw sequence. We mixed the raw sequence reads into various premixes, and then just ran it through a bunch of software that's out there. We had different lengths of sequencing reads, different sequencing depth, different proportions of taxa within that, and different overall number of species. Our highest number was 426 because that's the maximum number of species in the oral microbiome database. And then we ran it through a bunch of different pieces of software. In our simulation studies, this is the true positive rate on the y-axis and false positive rate on the x-axis. So you want to be up here in this corner with low false positives and high true positives. And in fact, this is where Pathoscope is. This is Pathoscope, the best performing method of the ones we tested. This is Kraken, which is very close. They do something fairly similar to us. And it's fairly close in performance, although not a little more false positives and a little less true positives. But some of these methods are just kind of all over the place. This thing called Taxator is way out here with a ton of false positives. So methods vary quite a bit in their performance, and we can even look at 16S. Chime, which you're going to hear about later with Greg, is a great piece of software and works well on 16S. And this is not so much a limitation of the method, well, a little bit of a limitation of the method, but also the data, because 16S is just, it's good at these higher level classifications, but when you get down to species level, I mean, for some species, 16S just doesn't vary. So, but when we looked at the same data, the same 16S data with Chime 1 and Chime 2, and then Pathoscope with either a green genes database, our whole genome database, or RefSeq database, at these higher levels, we perform pretty similarly. Chime 2 seems to have a lot more variance, but otherwise, in terms of the accuracy, they're all pretty accurate, although Chime starts to drop in accuracy at the ordinal level, and then at the familial level, and at the genus level, they kind of all are dropping. Even at the family level, Pathoscope is dropping as well, below the whatever that line is, 85% probably. But then at the species level, even Pathoscope is not doing great, although it does much better with whole genome reference data or the RefSeq library. But then when you use green genes as the reference library, you get significant reduction in the accuracy, less than 25%. So using Pathoscope with the RefSeq library, even with just 16S data, you're still doing much better than a Chime approach. And the fact that Pathoscope performs poorly with green genes suggests that this is probably a reference database issue with the single gene. And Pathoscope can take advantage of having a broader data set with the RefSeq library. So Pathoscope performs nicely. So let's go ahead and use it. Let's go back to that pediatric asthma study. So we have kids where we sample some snot. The traditional way is to take that swab and streak it out on a plate and look at it under a microscope and figure out what's there. We don't have to do things like that anymore. That's 100-year-old science. We can do metagenomics, shotgun metagenomics, map those reads, and then identify what's going on. When we did that in this pediatric study, we've got cases and controls. We noticed a big difference, principally in these big, whatever color this is, band, which correspond to Moraxella catarrhalis. So we got species level information. We, being good bioinformaticians, didn't know what this thing was. But we know how to use Google. So we Googled it, and and our colleagues said, yeah, that's, or the Google said that that's associated with upper respiratory disease. So we called our physician colleagues at Children's National and told them that, we found Moraxella catarrhalis, and we think that's probably what's going on. And they said, that's great. How do you know that's actually causal? And we said, well, we'll discuss that and get back to you. So we went back to the drawing board, and we realized that even though our raw data suggested that there's this Moraxella catarrhalis, this is RNA-seq data in this case. So the beauty of our approach is you can use it with DNA sequence data or RNA-seq data. We had 6 cases and 8 controls. We can go back to the first thing Pathoscope does is it takes all those sequence reads, in this case RNA-seq, and it maps them against a human genome first and sets all the human genome's data aside, and then surveys the pathogens. And depending on what kind of microbiome you're sampling, right? If you're doing a gut microbiome, our experience is that the gut microbiome, when you extract DNA and do sequencing, on average, you have about 50% host reads and 50% reads from that are microbial. With nasal swabs, you actually have 90% of the reads that are from the host, and only 10% from the microbiome. So this is categorizing 10% of those reads. So you have to sequence a little deeper sometimes, depending on where you're sampling. But we thought, because it's RNA-seq, we can actually survey the host, right? This is my son, who's now a 17-year-old high school kid. But we can go in and sequence or survey these 90% because it's RNA-seq and look at gene expression that's going on. And so we found a paper that had identified a whole list of candidate genes that were associated with Moraxella infection. So we surveyed those candidate genes and looked for differential gene expression in the asthmatics versus the controls. And in fact, we found a lot of them that were significantly overexpressed in the asthmatics versus the controls. So it looked like in fact, the patients were mounting an immune response, the asthmatics. So we thought, then we called back our friends and collaborators at Children's and said, we've got now data to show that there's actually an immune response going on with these individuals that are asthmatic that is specifically associated with Moraxella infection. So we felt better about that, and then went ahead and published the paper. One of the tricks with these kinds of data sets is be careful because you need some computational power, because they can get very large, right? You can end up using up a lot of hard drive space storing these data. And it's not just the raw data. The metagenomics data itself uses up a lot of space, but then every analysis you do also generates data files, and some of them can be quite large, and you end up becoming good friends with your local IT people, because they quickly wonder what you're up to when you start taking up all the hard drive space on the computing cluster. So that's a regular occurring thing for us, and we finally convinced our university to invest in a bigger, better high performance computing clusters so that we have more space to do these sorts of things. But when you do the RNA-seq, it's especially powerful. But of course, the downside is it's especially expensive. So you can do the targeted amplicon sequencing, like a 16S study, on the order of $25 a sample. The metagenomics DNA sequencing is more like $150 a sample. And then when you do the RNA-seq, it's more like $300 a sample. So the escalation in cost is severe. But the benefit of doing the RNA-seq data is that you get functional information, not just direct functional information, not inferred functional information from the bacterial taxonomic distribution, but real functional information from gene expression. And you get that from both the microbes and the host. So it's really powerful data and gives you really nice insights into what's going on with these studies. So my dream study is to do the DNA shotgun metagenomic data and RNA-seq data and metabolomics data all from the same set of samples. And I haven't found anybody to pay for that yet. So we're still looking, but that's the kind of dream study that we go for. So let me show you another example, and this one's more in the natural history realm. As I said, we collaborate with a lot of different folks from a lot of different areas because microbiome is important for a lot of different areas of health and behavior. One of our collaborations is with folks at the Smithsonian at the National Zoo, where they have black rhinos in captivity. And black rhinos in captivity have this issue with iron overload. And they're not quite sure why, but they suspect it has something to do with the gut microbiome that leads to obesity, that leads to some insulin resistance that creates this feedback of iron overload. So they end up being diseased in captivity. So we wanted to we actually want to do a more robust study. We've got funding now to actually sample these captive versus wild animals and do metabolomics. We're actually collecting the metabolomics data now to add to our microbiome data. But the first initial approach was just to look at the microbiome of black rhinos in captivity versus wild rhinos. And this is collaboration also with folks at Kruger National Park in South Africa. That's how we got the wild rhinos. So we had 17 wild rhino fecal samples and 8 samples for captive animals. So as I said, we're adding data from metabolomics now to go with this, but we're also adding more samples. We've got additional captive samples and additional wild samples, but our initial study looked at 17 wild and 8 captive. The beauty of black rhinos is when you're looking at gut microbiome, there's no shortage of material to work with. So this is easy sampling. The downside is while there's plenty of material for the wild rhinos, it's actually difficult to get that material. You can't ship that material from South Africa into the US to do sequencing. So I happen to have a former postdoc who's on faculty at Stellenbosch University in South Africa. So we were able to have the veterinarians at Kruger National send the fecal sample to my former postdoc who extracted the DNA for us, and then the DNA ships just fine. So we got the DNA shipped to us, and we did the metagenomic sequencing and ran it through Pathoscope and did some statistical tests. And you can see here are wild animals and here are captive animals, and you can see that they're very different in terms of diversity. They're very different in every respect. The wild animals were more taxonomically diverse. The captive animals basically looked like you might expect, like a horse gut microbiome. So because that's seemingly what they're feeding rhinos in captivity, food that looks a lot like whatever you feed a horse. So we're now thinking about alternative feeding strategies and even potential probiotics and things like that to help diversify the gut. So there are a lot of reasonable and important impacts from these kinds of studies that can inform downstream intervention strategies. So we're continuing to do this. We had another study. This is more a human study, a fecal microbiome transplant study that we did with collaborators at Innova Health here in Northern Virginia. The concept is that you have kids. This was the first fecal microbiome transplant study in a pediatric context. You had kids who were had already suffered from C. diff infection. And so they were at high risk for subsequent C. diff infection. They were back in the hospital. And so they wanted to do an FMT to basically try and prevent C. diff infection in these kids. So we had 9 kids who underwent C. diff infection. The donation was the donor material. If you don't know about fecal microbiome transplants, it's just what it sounds like. There are different ways to get the donation. In this case, they got the donation via colonoscopy. But there were different donor materials. So some individuals receive donor material from relatives, others from commercial donor material. Unfortunately, with only 9 kids and 2 different kinds of donor material, we didn't have enough of any single class to look at differences between the relative donations versus the commercial donations. But that's one of the things we want to look at in the future. We did our metagenomic analysis. The nice thing about this study was that we took longitudinal samples, so we could look at what's going on over time. We could look at what the starting point was, and then what happens after, and the starting material, and then what happens over time, to see if diversity changes over time, see if there's a difference in C. diff infection. And if there's a potential for pathogen passage. This has been a big issue with FMT studies. In fact, there was a death from one of these studies in 2019 in the U.S., which prompted the FDA to put a moratorium on fecal microbiome transplants till they figured out what's going on with this. So there's a lot of interesting basic science in FMTs, but there's also some interesting regulatory science in terms of fecal microbiome transplant and what you're actually transplanting, right? What you're moving. Are you moving pathogens? Are you moving antibiotic resistance genes, et cetera? So our data were already collected, so we could continue and analyze our data. So we have our donor material, and then this is the diversity of these kids before the transplant, and you can see very different from the donor material at the start, and very low in diversity. And then 2 to 7 weeks after, 8 to 13 weeks after, so 4 time points after the donation, and you can see that immediately the donation, the kid's microbiome looks a lot more like the donor microbiome and is a lot more diverse, and that diversity is maintained over time, and that's what we were looking for. Importantly, it also prevented C. diff infection. So these kids did not get any C. diff infection. So that was the goal of the study. We could also look at pathogens, and in fact, there were pathogens that we could identify in the donor material, most of which were, some of which were successfully, you might say, transferred to the kids. So that is a real issue, and we have to be careful about what donor material we have and survey that material for pathogens before we do the donation. Here it was all post hoc, so we couldn't do anything about it, but it's clear that some of the pathogens were transferred and others petered out. There's also, before the transplant, these kids, because they were on antibiotics, which is why they're their gut microbiome was relatively low in the first place, and why they were susceptible to C. diff infection. They had a lot of antibiotic resistance genes in their microbiome. The donors didn't have any. And after that donation, you really cut down the frequency of these antimicrobial resistance genes, which was important. So the upshot is you really have some powerful approaches. The FMT seems to work. There was one individual that had that did get the C. diff infection, but by and large, C. diff was kept at bay. So the FMT seemed relatively successful at blocking C. diff infection, reduce the antibiotic resistance genes in the gut microbiome of these kids, but did also have some impact with the transfer of pathogens. So the gut microbiome, it turns out, is also a fantastic tool for education. And a lot of this work that I'm presenting is collaborative work with a colleague of mine at the Computational Biology Institute, Marcos Perez Losada. And Marcos teaches our genomics course at GW. And he was taking a workshop on teaching and came to me and said, you know, we're talking about doing a project-based learning kind of thing. What do you have any ideas for what we could do for genomics? And I said, yeah, you could test the grandma hypothesis. And he looked at me and said, what are you talking about? What's a grandma hypothesis? And I said, well, this is my grandma here. She was born and raised in Arkansas, Indiana. She's 1/8 Cherokee, Native American. And my parents, these aren't us, but this is just off the internet, but my mom somehow didn't have a picture of the three of us in a tub. But I have an older brother and a younger brother, and my parents would dump the three of us off on my grandma in the summer for a couple of weeks. And grandma, being a great grandma, would take us out and get us all muddy and dirty. You know, we'd go fishing and tramping in the woods and just do all kinds of stuff. And then she'd bring us back and throw us in the tub, right? And she'd always tell us, make sure you wash behind your ears, between your toes, and your belly button, right? So these are what I suggested to Marcos were grandma hotspots, right? So these are the places that grandma thinks you need to really focus on because you don't normally hit those very well. And so I said, why don't we test the hypothesis that that those areas are somehow different. The microbiome of those areas are different than normal areas. So we decided we could sample a forearm and a calf as two normal locations in the human body, and then between the toes and behind the ears and in the belly button as the grandma hotspots. And Marcos developed this great approach to looking at this hypothesis in his genomics class, where he could carry this study out throughout the entire semester. So start with a lot of formative assessments for each step, with a summative assessment at the end where the students actually write a report and report the entire study. And it starts with basic information on sampling, right? How do you do sampling for a microbiome study, for any kind of scientific study, but for a microbiome study in particular? How do you do sterile sampling? How are we going to sample our 5 sampling locations? You know, get a lab notebook, start writing down everything like we do. So the students actually sample themselves and put the samples in solution. Then we do this in a wet lab classroom, so they can take those samples, extract DNA in a different week when they learn about DNA and DNA extraction and gel electrophoresis. They can run their DNA out on the gel and show that they have DNA. Then they can do a PCR amplification. So here we just do 16S because it's an educational activity, and we have no budget for it, so we can't afford a lot of money. But you can do the PCR reaction and then run that out on a gel and show that you've got the PCR product you want. Then we take that to our DNA sequencing facility, and they do the targeted amplicon sequencing and provide us with sequence data. And then the students get into the bioinformatics, right? So we have a bioinformatics pipeline where they run the data through a variety of algorithms to analyze the data, measure relative abundance, and then throw it into Microbiome Analyst and do some statistical testing. And then they can write a summative report. So it's really a nice way to do this. We've actually published this whole process in the Journal of Microbiology and Biology Education. So we've got very explicit step-by-step instructions on how to do all this. But the fun thing was we actually, Grandma was right. Grandma was right. These grandma hotspots, ear, navel, and toes, are actually less diverse than calf and arm, right? So presumably they're being overrun by a particular or by a few dominant species, but there was a significant difference. We actually expected them to be more diverse, but it makes some sense that they're less diverse, but definitely significantly different than the control areas of the arm and the calf. But it's the other interesting thing is that Marcos had run this class without this project-based learning activity to run through the whole semester. And then he ran it again with the project-based learning. And there are a couple of direct assignments where you can look at performance, and the students did significantly better with the project-based learning, as you might imagine. You know, when you're doing hands-on stuff, you're more invested and you actually learn better. But they also enjoyed the class better, right? So they had more positive experience that they reported with the class at the end of the post-course evaluation. So, you know, the education was significantly different. The grandma hypothesis was significantly different. And we've had to shut this exercise down with COVID, but Marcos teaches this in the spring, so he'll be teaching it next semester. And we're ramping it back up. So we're hoping we think it'll actually be really fun to repeat this for a couple of years and collect an abundance of data and see if this holds true year after year. So we're excited to continue this effort. Finally, I want to talk about another piece of software that allows us. It's in our Pathoscope suite of software. This one's called Telescope. It specifically looks at transposable elements. So transposable elements are another component of that microbiome. And in fact, transposable elements make up 45% of the human genome. They were originally discovered by Barbara McClintock, who won the Nobel Prize in Medicine in 1983 and is still the only female to win that Nobel Prize unshared in that particular category. She discovered these transposable elements working on maize. Transposable elements are what give corn this diversity of color, the transposon activity. Of course, transposable elements were those part of the junk DNA, which is a term we use for stuff we don't understand. But it turns out a lot of these transposable elements, some of the transposable elements are historic evolutionary artifacts that are non-functional and just hanging out in your genome, but some of them are actually expressed. And the expression of these transposable elements can disrupt normal function of downstream genes. And in fact, this transposable element function has been implicated in a variety of human genetic diseases. So we wanted to take a look at these transposable elements and with specifically with cancer. And so we developed some software to take a look at these transposable elements called Telescope. And you know, it was a very familiar problem after Pathoscope, because you basically have unique reads and you have ambiguous reads. And most people do what I said before, either throw away the ambiguous reads or lump them with the unique reads and get low resolution. So here, though, instead of just taking all sequence reads, we're specifically looking in the RNA-seq data for the hallmarks of transposable elements, so which are 5' 3' LTRs with some open reading frame in the middle. So that's what we're targeting with telescope. That was our adjustment relative to pathoscope to allow it to specifically pick up transposable elements. But we still use the Bayesian reassignment approach to help these ambiguous reads ride along with the unique reads and give us better resolution. The nice thing is, because you're mapping these reads against a human genome and identifying specific transposable elements, you also get specific chromosomal location for when you get hits. So not only are you getting expressed transposable elements because it's RNA-seq data, but you know exactly where they're being expressed in the human genome. So we specifically targeted human endogenous retroviruses, HERVs, to look at their impact on cancer. And we've done this with a couple of different cancers. So we use the TCGA, the Cancer Genome Atlas data, because it's a beautiful data set of RNA-seq data that includes normal and cancer paired tissue from which RNA-seq was collected. So it's a great data set. And you can run it through telescope, and you can immediately identify a bunch of HERVs that are differentially expressed. Some are underexpressed, some are overexpressed. When you look at these differentially expressed HERVs, they partition out between tumor and non-tumor tissues tremendously well. We found over 3,500 expressed HERVs. Over 1,000 of them were differentially expressed in this head and neck cancer data set. So they make a difference in terms of survivability, right? So they're clearly impacting survival with head and neck cancer. And because we have locus-specific information, we can actually map them specifically to locations on human chromosomes. So this is a map of the human genome in a circular way, with each one of these being a different chromosome. And then on the outer rim are all the HERVs that we found that were expressed, with the red ones are the differentially expressed HERVs. And then the inside the circle are the genes. Because it's RNA-seq data, you can also survey the differentially expressed genes. And then you can start looking at associations of specific HERVs with specific genes. And we've done that now with a few other cancers, similar kinds of data sets: breast cancer, prostate cancer, TCGA data, and found similar sorts of things, large volumes of differentially expressed HERVs associated with specific genes. We're now developing software to sort of isolate these associated genes a little bit better. And we also have collaborations at GW with our cancer center researchers to go in and do some lab work where we can express HERVs in front of particular genes and look at the expression patterns of those genes and see how those HERVs are being expressed. But we're also developing additional software to take this another step further with respect to the host genes and the HERVs and their differential expression, and using some machine learning approaches to identify specific significant associations between these, and then look at the pathways that are associated with those using some deep learning approaches, deep path, to identify particular pathways that are being impacted by those. So this is a collaboration with Ali Ranavard, who's also a faculty member in our Computational Biology Institute, and his software suite called Omics I. So we're actively developing these kinds of approaches, and these are generic approaches for multi-omics data integration. Here we're using expression data from host genes and from HERVs. So output from telescope, output from Maslin 2 or DNA-seq or something like that, and then looking at what's going on. But this software is fairly generic, so you can look at any omics features. So we're using the same kind of software to look at metabolomics and how metabolomics are associated with different differentially expressed genes, as well as differentially expressed HERVs. So it's really robust software that allows you to look at all kinds of different omics data and identify significant associations and identify pathways that are being impacted. So, of course, this is all done by a team of fun and exciting collaborators. Evan Johnson helps us with the pathoscope development. Matthew Bendall is a former student of mine who now works in Doug Nixon's group, who's developed the telescope software. Ali Ranavar develops the Omics Eye software. Rob and Marcos are associated with the asthma study, really ran that. And of course, Marcos is also the one doing the educational genomics, educational microbiome project-based learning. Keely Gibson and is a former graduate student who worked on the rhino study, and Buddha is our colleague at the Smithsonian. Michelle Ahn was a master's student in the group who worked with Suchi, who's a collaborator from Innova Hospitals, and Maria is a collaborator at Johns Hopkins. And of course, this stuff can't happen without some funding. So we've got funding from Innova, from NIH, from the Rhino Foundation. And we even convinced the local brewery, whose name is Lost Rhino, to give us some in-kind support for our Black Rhino study. We had a little Black Rhino party over at Lost Rhino, and they gave us some good beer to drink. So metagenomics allows for exceptional characterization of both the microbiome and the host at both the taxonomic and functional levels. So you can use metatranscriptomics, RNA-seq data to really get at functional. You can use the metagenomics or targeted amplicon sequencing to look at the taxonomic diversity. Really powerful approaches these days to characterize microbiome diversity and then test for associations with other phenotypic characteristics of interest. Our pathoscope software is useful for a variety of analyses. We're actually developing a newer version of this called Metascope. It's just about ready for public consumption. So we've got more activity on this, and it's coming. Telescope also opens this door to the retrotranscriptome. So what's going on with these transposable elements? And they seem to be very impactful in a wide variety of diseases. I showed you a couple of examples from cancer, but we've also used this approach to look at what's going on with lupus and autoimmune disease. We've looked at it in mental disorders like schizophrenia and depression. And we find, wherever we've looked so far, we found transposable elements actively transcribed, being expressed, impacting human health. So it seems to be a pretty powerful and broad tool for looking at what's going on with respect to transposable elements. And then the Omics Eye is this platform for multi-omics data integration. So we can look at the genomics, the transcriptomics, add on metabolomics, proteomics, use these powerful machine learning techniques to integrate all this with the clinical data, right? With the metadata, the clinical information, and really do some interesting science and identify significant associations. And so, as you might imagine, with all this stuff, informatics becomes an essential skill for any biologist. So we do a lot of informatics teaching. We have new master's programs, a master's program and a PhD program in health and biomedical data science through our Department of Biostatistics and Bioinformatics. Each of those programs has a track in bioinformatics and a track in biostatistics, and both draw from each other significantly. So if you have a background in biology and you want more informatics, this master's program is a great opportunity to advance your skills in informatics. And if you have those skills already and you want to go on to a PhD, our PhD program is I think fairly unique in terms of the combination of informatics and statistics that we do. And we have all kinds of great research projects to work on. Our collaborators in biostatistics run our Biostats Center at GW, which is in Rockville, Maryland, and they do all kinds of NIH clinical trials. So really great opportunities for biostatistics and bioinformatics at George Washington University. So if you're interested as students out there, I certainly encourage you to think about applying to programs. We've got our software is all on GitHub. It's all open access. So go find it, have at it, have fun with it. Don't email me if you have issues. email Evan if you have issues with Pathoscope. Email Matthew if you have issues with Telescope, and email Ali if you have issues with Omics I. I learned a long time ago. I'm an old enough bioinformatician. I learned early on that software maintenance is not particularly fun, and dealing with all kinds of issues that people have running your software and their unique computing systems is not necessarily fun to deal with. So I like to conceptualize software, help people build it, and then have them take it and go away with it. And that's what's happened with all these pieces of software. So I'm just happy to be able to continue thinking about how to improve the software, helping improve it a little bit, and then applying it to a lot of interesting studies. So thanks for the time. We are the Computational Biology Institute at George Washington University, and you can tweet me at Crandall Keith, email me at kcrandall@gwu.edu. Our Facebook page and our website, frankly, are woefully out of date because, like a lot of places, our university fired everybody who did things like that for us. So with the COVID, so we're hoping to revamp some of this here quickly as we're coming out of our financial crunch and back into reality. So I'm happy to take any questions at this point. Hope that was helpful.
Thank you so much, Keith. And first I'd like to apologize because I was speaking for nearly 20 minutes and I didn't notice that my electricity was out. And I continued. I even made jokes, which I'm very,
Did you give my introduction?
I even got through the introduction. I talked about our innovation lab where I met you and everything, and nobody hear a thing. And when I finally got the electricity working, you were already speaking. So I would like to apologize to you and all our attendees for this technical mishap.
No worries.
So before I go into questions, because we still have a lot of time, we have at least 20 minutes. We are 20 minutes ahead of schedule. I'd like to introduce you. And I'm not going to go over my slides anyway. They were very, very cute, very cool, actually. But I'm not going to go over them. I'd just like to, give an overview. People already saw the amazing work that you do. It ranges from rhinos to kids' health, which is pretty amazing. So I'm going to read your bio. First, I'd like to highlight that you're a AAAS fellow. I'm actually now the president of AAAS Caribbean Division. So for us, it's an honor to have you in our session. Dr. Keith Crandall earned his BA from Kalamazoo College in Biology and Mathematics. And it's obvious that you have a very strong background in mathematics and statistics. A master's degree in Washington University in statistics, a PhD from Washington University in biology and biomedical sciences. He's a computational biologist, bioinformaticist, and population geneticist at GW, where he's the founding director of the Computational Biology Institute and the professor at the Department of Biostatistics and Bioinformatics. Professor Crandall studies from computational biology of crustaceans to agents of disease. His lab also focuses on the development and testing of big data methods and DNA sequence analysis, applying methods to studies of the evolution of infectious diseases with a particular focus on HIV evolution, which you haven't mentioned today. Professor Crandall has over 270 peer-reviewed publications, several books. The number of publications is really amazing, and he has been also highlighted as the Institute for Scientific Information has one of the most cited scientists. Dr. Crandall's research has been funded by the National Science Foundation, the National Institutes of Health, National Geographic, U.S. Forest Service, so you name it. A lot of institutions have funded your work and supported you. He is also a Fulbright visiting scholar to Oxford University, and he was also on a sabbatical fellowship at the University of Auckland. He received a number of awards, including the Alfred P. Sloan Foundation Postdoctoral Fellowship, the American Naturalist Society Young Investigator Award, an NSF Career Award, and the James Shannon Director's Award. He's also a E.O. Wilson Naturalist Award, which is for a computational biologist is a big honor. So
well,
anyway,
that was not for computational biology. That was for my.
No, I know, but it's really amazing. So yeah, you have shown today that data is a very getting to work with data and understand and apply statistical methods to sequence data is an important skill to any microbiologist today or any biologist today. And you also have shared some of the opportunities at your institution, which frankly we have to discuss ways to send our students even for a summer to get training in all these tools like telescope and the multi-omics integration. We are very interested in learning these skills. So yeah, we have a couple of questions now, and I don't know if they want the floor, but Marco Cardin has two questions. The first is, can you report microbial relative abundances when using statistical models, such as crossover design, where data is transformed? That's one question. The other is also regarding data. It's about depth of sequencing shotgun metagenomics of the gut microbiome. What sequencing depth do you suggest?
So the first one's relatively easy. Yeah, you can calculate relative abundances pretty easily. Our software doesn't do that. Our Pathoscope software gives you basically a CSV file as an output with read counts by GenBank taxa IDs, right? And you can, the nice thing about Pathoscope is it will get you down to strain levels. So you can differentiate strains of E. coli, for example, and have read counts for each strain. But then you can take those read counts and throw it into other downstream analysis packages, which we always do, and calculate relative abundances, do normalizations and all that stuff. So like anything in bioinformatics is not about a single package or even a single platform. It's about lining up 20 different tools and trying to figure out how to get the data, the output from one tool as input to the next tool, because somehow they never talk to each other very well.
Exactly. And I was wondering myself. So we tried to follow the international guidelines to be able to do meta-analysis with other studies. So we deposit our data into Cheetah, and Cheetah is now enabling metagenomic analysis as well. So could we download from Cheetah and input into Pathoscope for this species level analysis that you're mentioning? Or
if you're downloading raw, I mean, the input for Pathoscope is just raw FASTQ files.
FASTQ. Okay, so we would have to input from the start. Okay.
Yeah. But the other question is an interesting question as well, and that one's one that I deal with all the time, because in addition to running our Computational Biology Institute, I also run our genomics core. So anytime anybody wants to do a sequence analysis, that's always my first question to them, and they of course have no idea about sequencing depth or what that means, or they just have samples and they want sequence data. Okay, but is there a question, and what is your question, and that will dictate how much we sequence. But it's also interesting, especially the microbiome context, because as I kind of alluded to with the asthma study, when you do, for example, a nasal swab, 10% of the sequencing reads are microbial, whereas 90% are human. But if you do a fecal sample for a gut microbiome, it's about 50-50. So And we're working on a study right now on breast milk, and breast milk is like 99% host and 1% microbe. So what we usually target for metagenomics for microbiome is somewhere between 2 to 4 million reads per sample of the microbial population. So to get 2 to 4 million reads of the microbial population, it depends on the sample type. And because of this weird change in proportion of microbes to host. So you really have to, you have to understand, you have to have some a priori knowledge about that. And if you don't, then our suggestion is always sequence as much as you can afford, and then we'll figure out what's going on. And
Yeah, but I think we should also mention that there are also pre-sequencing methods which you can enrich your microbial
right.
sample, so you can deplete the host signal
Deplete the
before
host.
sequencing.
Yeah.
And that's very useful.
And that's kind of an underappreciated reality to a lot of the microbiome work, is how some of the sequencing steps can really impact what you see in the microbiome. With that asthma study with Children's National, for example, we reported back this Moraxella that seemed to be overabundant in the asthmatics. And our collaborator, Rob Frystad, his first reaction was, where are all the viruses? Because this is kids' snot. It should be jack full of viruses. And it's one of those, we'll get back to you, right? So we scratched our heads, we sat around and we thought, well, let's just test our pipeline and throw in a bunch of viral short sequences into our data that we already had, and titrate those sequences, basically do a dilution experiment, right? Where you throw in different quantities down to very small quantities of viral sequence, and then run it through Pathoscope and see if we identify it. And we could identify it even at really small frequencies. So we went back to them and said, we're pretty sure it's not on our end. How are you guys prepping the samples? And it was on their end. They did a prep that eliminated all the viruses. so your DNA extraction approaches, your library construction approaches, some of these different molecular lab techniques can really impact what you are seeing downstream. So you really have to be aware of every step of the process. Even the sample itself, right?
The way it is stored.
We're working on these rhino samples right now, right? And it turns out that rhino poop has a lot of grass in it. And that kind of gums up your DNA extractions to a certain extent. And it's like, how do we deal with all this grass? We're not quite sure. So it's always a trick.
Yeah, definitely. And there are a lot of protocols, and this comes to answering questions by Dr. Ricardo Gonzalez. When doing these complex pipelines, could you comment on how you approach on reproducibility of the results in robust experimental design, particularly applied to clinical data? And just for saying, there's the Human Microbiome Project and there's the Earth Microbiome Project with specific protocols for all types of samples, with catalog numbers of things that you could do to reproduce, the gut, skin, vaginal, all these samples. You can follow these protocols and you can do meta-analysis in the end, right? So can you please comment on this? And then I'll go back to the other questions I have.
And this is a big area, frankly, of research, is how do we worry about these kinds of approaches, especially for clinical applications. And before I got to DC, I thought NIH and NSF and FDA were the government organizations, right? But I've since discovered that here in Washington, DC, there are all kinds of other government organizations, one of which is something called NIST, the National Institutes for Science and Technology. And these are the people who apparently define what a gram is, for example. And they are very interested in microbiome studies and very interested in exactly this question of how do you standardize both the lab approaches and also the computational pipelines so that you can get reliable, consistent results that you need to do clinical applications. So they have a whole working group, which I'm part of, on microbiome studies. And we've been going at it for a couple of years now, and we're still a long way from having standards. But that is the goal.
And please remember that people around the world from countries that don't have ultra minus 80 freezers, in Africa people are doing microbiome research. In South America, in the jungle, people are collecting fecal matter from Indians with high diversity guts in order to store them for humanity. So, and every one of these guys needs to be able to use these methods. So we have to not only think about the first world and how the US and Europe does work with the microbiome, but also extend this and being more inclusive, doing the microbiome field more inclusive.
And in fact, the first part of our pipeline is mapping these reads to a human microbiome or a human genome, so you can extract the human reads and then focus on the microbial reads. When we do that, we've done another collaborative study with Children's National where they're sampling folks in the Congo who suffer from a disease from undercooked cassava that does this neural paralysis thing, and the human genomes that are available for mapping reads against are not from the Congo. And so, these reads, 50% of the reads you can't even map to anything. And that's both a lack of understanding of human genomic diversity, as well as a lack of understanding of, frankly, the gut microbial diversity from people who are not from Western North America eating McDonald's, right? So we've, the Human Gut Microbiome Project was a nice effort to get us started, but it's just a start. It's just a start. There's so much. I tell people in the microbiome world of science, we are Darwin in the Voyage of the Beagle. I mean, we're just sailing around collecting stuff, trying to figure out what the hell's going on because we have no idea. And I can't tell you how many times I've gotten the review, this is descriptive science. Well, yes, it is. Because guess what? You have to describe some stuff before you can start testing hypotheses. You have to have a baseline knowledge. And we're not even there with the microbiome.
Yeah.
We don't have a firm understanding of what even just the gut microbiome looks like in different populations of humans around the world. We certainly have no idea what the gut microbiome, how the gut microbiome changes across a lifespan. We have no idea how it changes just temporally, right? When you how it changes with respect to what you're eating. How does it change when I travel to Mexico and start eating some famously hot salsa? I mean,
yeah.
I'm guessing that impacts my gut microbiome, but I'm not sure how.
That's why it's so essential that now we can train students and we can train even physicians to understand how important is this. And in order for, in a couple of years, 5-10 years, even physicians can talk about this with their patients. And I think it's going to be a reality soon. So we have one last question here from Dr. Jorge Miranda. Probably I missed this. Did you find any difference between the commercial versus relative donors in the fecal matter transplant study?
There were some differences. They were not statistically significant just because we had low sample size. So that's definitely one of the things we want to go back to with a bigger sample and look at more intently. So there were hints of some differences there.
Yeah. So I have another question from a student, Eduardo Tosado. He's a PhD student. He asks if you can combine different sequencing technologies like Nanopore, Illumina, PacBio, Illumina with Pathoscope.
You can. We principally look at like I said, the input is FASTQ files. So whatever you can get into a FASTQ file. The interesting thing is all this software has really been developed. All the software for microbiome analysis has been developed for short read sequence data.
Yeah.
With the long read data, it's not. We've done a few simulation studies to look at longer reads, and frankly, I think these methods don't perform particularly well, and they certainly are not geared towards taking full advantage of the long read sequence data. Incorporating their error profiles and things like that. So I think there's a lot of opportunity to expand these kinds of methods for long read data from platforms like PacBio.
Yeah. So I have one last question before we leave to the second session. Those teaching laboratories where students collect their samples, it's really cool. I had developed in my former institution a microbial ecology course where I asked people to bring water, and we extract the DNA, and we sent the sequence along with my project sequence data, and we analyzed data for the rest of the semester.
Yep.
Obviously, we didn't publish. We don't have the resources you guys have there. But I was wondering, to do that on self-collection, you need an IRB for that course, or it doesn't really matter? It's just a sequencing experiment. Do they actually require you to have that?
I'm trying to think.
Out of curiosity.
Marcos says we do. So.
Okay, nice. Yeah, I was wondering about that. Anyway, Keith, amazing talk. You covered, everything. Again, I apologize to our audience and to yourself for this technical mishap. I'm gonna ask you to leave the door. There's a door sign in the left bottom, and then enter the second session for Dr. Alan Landay. Again, thank you so much, Keith.
All right,
have a great day.
Thanks, everybody.
Bye.
Okay, I'm hearing the music for some
Music
reason.
is still playing though.
Okay, perfect. Okay, good morning. I am Gabriel Borges, a student of the doctoral program of Microbiology and Medical Zoology Department at the University of Puerto Rico Medical Sciences campus. Today, I am going to be the moderator for this session. I am going to be giving you some guidelines for everyone to enjoy this session at its fullest. All participants who are in the session will be able to listen and view the speaker and moderator, as well as the presentation of the speaker. All microphones and webcams from the attendees of the sessions are not visible. During the question and answer session, attendees interested in asking questions and appearing next to the speaker of the session will need to press grab the mic at the bottom left of the screen. The production will grant access on first come, first served basis. Grab the mic won't be granted by production while the speaker is giving his or her talk. During the question and answer session, questions can also be written in the chat. The 5 minutes for question and answer will apply to both questions asked live and questions written in the chat. Once the session is over and for attendees to access the following session will need to click on open door icon at the bottom left of the screen of the current session. Once you see that the agenda, click on preview icon that appears to the right of the session title to access the session. If the session has not yet started, a timer and background music will appear until the moderator and speaker or production are in the session. Important disclaimer: the content of the session and all other sessions cannot be reproduced without prior authorization from the organizer and the production. Okay. Today, it is an honor for me to present Dr. Alan Landay. Dr. Alan Landay is a professor in the Department of Internal Medicine and Microbial Pathogens and Immunity, and is vice chair for research in the Department of Internal Medicine at Rush University Medical Center in Chicago. He has been involved in HIV research for over 40 years, having performed some of the first immune evaluations of HIV-infected hemophiliacs in 1982, while completing a postdoctoral fellowship at the University of Alabama Birmingham. He collaborated with Abbott Labs in 1983 in helping develop the first HIV antibody test that was licensed by the FDA in 1985 for use in screening the blood supply. His laboratory has made important original contributions in understanding the role of immune activation and inflammation in outcomes of non-communicable disease related to aging in the HIV population. This work has been translational focused with his participation in AIDS clinical group and leading clinical trials on interventional strategies to reduce systematic inflammation and activation in the HIV population. He has extensive experience in aging-related studies and is co-investigator of an R33 grant that integrates efforts of the Center for AIDS Research, Older American Independence Center and Chuck Center. He has participated on the Office of AIDS Research, HIV and Aging Committee, and has been an advisor to UN AIDS on HIV and aging. Over the past 10 years, his research focus has been on studies of the human microbiome and metabolome, and better understanding its role both in health and disease, resulting in over 40 peer-reviewed publications. He has been the chair of HIV Microbiome Working Group for Women's Interagency HIV Study and co-chair of the Microbiome Working Group for AIDS Clinical Group. He has chaired the International HIV and Microbiome Meeting sponsored by NIH for the last six years. uh, it now now okay, excuse me. Okay. Now I let you in the hands of Dr. Landay and its presentation titled HIV aging and the microbiome, a dangerous trio.
The music is
I
still
think you
playing.
may have an additional tab open, and that tab may be streaming the music.
Oh, there. Sorry.
There
Now
you
I
go.
got it. Thanks. Thank you so much. Are we? Yeah, go ahead. Sorry.
Give me a second. Let me. Uh, I have. Here we go.
Well, thank you so much for the wonderful introduction. I'd like to thank the organizers for inviting me. And we'll be talking to you today about HIV aging and the microbiome. I think a topic very much of interest to the audience, and having heard the really wonderful first speaker and the introductions, this will follow on with a lot of the same concepts. Next slide, please. What I will talk about today is some newer concepts around aging and understanding that we're now not looking only at lifespan, but we're changing the whole concepts of aging research to look at health span, and introducing a new field of science that's really been prominent at the National Institutes of Health around geroscience. We're moving from the consideration of longevity to considerations of health, and that you want to live a long age to your hundreds, but you want to live healthy. You don't want to live to 70 and then be in a nursing home for the next 20 years. And we now have the tools and the abilities to think about health span. When we look at the domains characterized in health span, we really look across all of human disease, and that includes neuromuscular, cognitive, cardiovascular, metabolic, inflammation, and body composition. And now there's a shift towards delaying and compressing the period of the lifespan when you're going to remain healthier, and therefore extending and improving health span. So these are some of the newer concepts I want you all to gain today. As students, these are areas you'll be working on and thinking about in improving health. Next slide. Next slide, please. Oh, thank you. When we think about aging and geroscience, which is this new field you may or may not have been aware of, it hypothesizes that aging is modifiable now. So there now is a modifiable risk for aging as we are all getting older. We think now we can intervene and deal with many of the chronic diseases of aging. And it's a modifiable because there are a limited number of pillars that are listed here on this slide, the 7 pillars of aging, that are involved in the aging process. And as we understand, we can intervene with these pillars, which I'll talk about today, that we can now modify aging and how along for a longer healthy health span in aging. And a number of these are things like epigenetics, inflammation, proteostasis, metabolism, and so on, things you've heard about that the microbiome, which will be a focus of my talk today, are also involved in. Next slide. So when I talk about inflammation as one of the main pillars, what are the common features of inflammation and immunosenescence and HIV and aging? And we now introduce another term called inflammaging. As you get older, there's an increase in the inflammatory responses that can lead to the senescence of the host, both the immune system and at the tissue level of the organs like the heart, the brain, the lungs, and so on. And as we're now looking at this, we think there are common features, both of normal healthy aging and HIV as a model, which I'll be talking about today. Next slide. These common features are involved in thinking about pro-inflammatory mediators, and those include acute phase reactants like C-reactive protein, interleukin-6, and TNF. And these form now the basis of the concept of inflammaging. So as you age, you increase inflammatory mediators. You also have elevated levels of coagulant factors, including things like D-dimers. So the coagulation system is altered during aging as well. Not only can we study soluble factors in the laboratory or on the clinical side, but we can also look at cellular markers. And these include important aspects of monocyte activation. And there are now what we call pro-inflammatory monocytes that are present during aging. These are the so-called CD16 monocytes. We can also look at soluble factors that are involved in monocyte or features of monocyte activation, such as soluble CD14 or soluble CD163. That soluble 163 is the hemoglobin scavenger receptor. So now we have laboratory tools, and as students, you can use these tools to characterize patients or even animal models to look at the questions of aging. We also know that there's an increased prevalence of other co-pathogens during aging, especially CMV. CMV is one of the important features of aging that drives senescence in healthy aging, but it's also present in HIV patients. Almost 100% of our HIV patients are found to be CMV positive, and it's this virus that we're now actually targeting to impact aging. We now have drugs and we have vaccines that we're using clinically in our trials even today to try to address how does CMV affect aging. We can now intervene and impact that. And we can then look at outcomes of these clinical features of dementia, cardiovascular disease, metabolic alterations, frailty, sarcopenia, which is the change in muscle function, and impaired immune responses. So all of these, and cancers. And so all of these are important clinical features that we want to modify during healthy aging. Next slide. So what about the microbiome? That's the real focus of today's symposium. And aging and the microbiome are really critically linked. One of the main features of driving aging is indeed the microbiome, but not the microbiome itself, but the products of the microbiome. And you heard from Dr. Crandall's talk about metabolomics. So it's now the adage in the microbiome field is we're not discussing who is there, but more what are they doing? What are the microbial products? What are the viruses? What are the fungi? What are they doing to contribute to these changes? Next slide. So you can see now, and we heard a bit about the gut microbiome, it is a stable community, 100 trillion microbial cells. And one of the fun facts for your next discussions or when you go off with friends is we are more microbial cells than we are human cells. And we can be at a ratio, it's been proposed at 10-to-1 or 5-to-1. So we have many more of these microbial than human cells that we're made up of. And you heard a lot about the developmental questions in the previous talk. We also have a knowledge that there's a role in nutrient metabolism and barrier function. So the microbiome is critical for maintaining the integrity of the gut. And what happens during aging, as I'll show you in a few moments, is the gut becomes leaky, and the products of the gut microbiome, those products that are related to lipopolysaccharide and others, are then found in the circulation and can lead to inflammation. We also know important roles that the gut microbiome is impacted by diet, sexual practices, and other factors. And it's also, as you can see on the left-hand slide, depends on where you actually take the samples. There are differential numbers of bacteria in the left colon, the right colon, the ileum, and the duodenum. So throughout the large and small intestine, the function of the microbiome and the number of microbial cells, and also the function differs. So again, like we say in real estate, it's about location, location, location. And we talked a bit about some of the analytic tools in the first talk. Here, we also talk, and importantly, about where do we sample and what kind of samples do we look at are critical for understanding the role of the host microbiome in disease. Next slide. So here you can see that there's an important role for aging and inflammation interacting, causing comorbidities. But many of the comorbidities on the right-hand slide here, where you see gut dysbiosis, an alteration of the human microbiome during aging, that can lead to changes in the nervous system, musculoskeletal, lung, in the gut part of the digestive system, also related to cancer and cardiometabolic. So again, there is a great important link between aging, inflammation, and the gut. These are very much linked. And as we think about altering the microbiome, which I'll come back to at the end, we can certainly affect aging and all these comorbidities of aging, or what we also call non-communicable diseases. And as I'll show today, HIV is another important factor that's involved as a disease that's prominent throughout the world as one of the first pandemics and epidemics I studied in my career now, and I'll follow on with some discussions of COVID-19 as well. Next slide. So when we look at aging, now this is just healthy aging, we find that microbial translocation increases in the elderly. In this case, we're using the biomarker of lipopolysaccharide, which is the outer part of the bacteria. And so what we can find in the elderly, as you can see on the right-hand side, that there's an increase in plasma LPS levels. The elderly individuals here are over 70, the younger individuals here are under 30. So during normal healthy aging, we find that the gut becomes leaky, and then there's a presence of endotoxin in the systemic circulation. This is now responsible for many of the inflammatory mediators in aging and can drive the comorbidities of aging and cardiovascular and other diseases that I've alluded to. Next slide. So what happens to the gut microbiome with age? We know there are, again, over the whole lifespan, there are significant changes in the host microbiome from the in utero phase all the way through to older age. And what happens in aging, and we certainly think about aging at a different level, usually in the field of aging and geroscience, we're looking at individuals in their 70s or 80s. But when we think about aging, as I'll talk about in HIV, we're looking at those individuals at a younger age, usually around 50, which is equivalent to what happens in healthy aging at age 70. So we find that there's a decreased stability in biodiversity. As we heard earlier, the gut microbiome is highly diverse, and that's a healthy microbiome. What happens in aging, you lose that diversity, and you have an increased abundance of gram-negative facultative anaerobes that are Enterobacteriaceae, and these have then pathogenic potential. You also then have a decrease in firmicutes and decrease of the firmicutes bacteroidetes ratio. Importantly, one of the functional roles of these firmicutes is to produce factors that are anti-inflammatory, namely short-chain fatty acid. And it's those short-chain fatty acid bacteria that are decreased during aging. And they're also decreased during HIV, common features. By losing these anti-inflammatory factors and the bacteria that make them in the gut, this leads to an increased leakiness of the gut, and then the presence of the bacterial products or markers of bacterial translocation in the systemic circulation. Next slide. So what happens is that there's common features, as I said, between HIV infection and aging. And we now are starting to think about HIV as a disease of accelerated or accentuated aging. And we can now, in the studies that we're doing in HIV, the age of these individuals is their 50s. And as you look across much of the US or Puerto Rico itself, we're going to find individuals mostly now in their 50s and 60s in the next couple of years with HIV who are living long and healthy lives, given many of the newer developments in antiviral therapy. But what we see then is an increase in the gram-negative bacteria with pathogenic potential. What we call these are good bacteria turned bad, and they're also now known as pathobiomes. One of the important features at the genus level are Prevotella. Prevotella are normal, healthy bacteria found in your gut. They're a normal commensal, but during disease states, and that's again an important feature, as students who begin to study what is a normal healthy microbiome, will those same bacteria that you find as healthy in the healthy state can become pathobiomes, and especially in diseases like HIV, these have been studied, like Prevotella, they can now become pathogenic in nature. And we also know then there's a decrease in bacteria, as I said, with anti-inflammatory properties that make the short chain fatty acids. So these alterations in the balance that changes both in HIV and aging, it gives us a target, which I'll come back to at the end, for interventional strategies, but also allows us to think about pathogenesis studies. Next slide. So where we know about HIV and AIDS is that they're independently associated with distinct changes in the fecal microbiome in older participants. Also, HIV infection status impacts age-related changes, and it's associated with increased soluble factors of inflammation, monocyte activation, and a pattern that's associated with microbial taxa in older adults. So again, there's an association that you change at the gut level, but then when you look in the systemic circulation, you can find inflammatory mediators. So as we say, what goes on in the gut does not stay in the gut. These changes in the microbiome then can be reflected by taking blood samples or looking across other sites in the tissue. And the most critical part of all of this is the changes in short chain fatty acids. Those bacteria that make those short chain fatty acid metabolites are lost. And that's what is one of the major contributing factors to this inflammation or inflammaging. Next slide. So what about other features or another opportunity to study the interactions of the microbiome? There's also now a new concept that's coming up called the glycome. And the glycome itself can have an important impact on the microbiome. Next slide. So the glycome, these relate to sugar structures that can either be found in the circulation, as you see here, or at the cell surface. And now we're beginning to look at the microbial glycome. What is the sugar structures on our microbiome and those sugar structures on the epithelial cells in the gut that can also potentially affect the outcome of the microbiome. So now we have a whole new field of study that's becoming much more prominent as another area to study on the interaction, both systemically and locally in the gut with regard to the microbiome. Next slide. When we look in HIV, we actually find it's associated with persistent alterations in the IgG glycome. Here we're looking at sialylated and fucosylated glycans. And what you can see on the far left-hand side is that you lose sialation, you have increased disialylated glycans as you go from an HIV negative to an HIV positive who is on effective therapy to somebody who is not on good therapy. So you're now showing a loss of sialylated glycans. And on the right-hand side, you're looking at fucosylated structures on IgG, which go up. And so then there's alterations in different parts of the glycome, either upregulated or downregulated, that correlate with disease state in HIV. Next slide. So our hypothesis is that HIV infection causes glycome dysregulation that persists and contributes to HIV-associated inflammaging and persistence, and maybe also associated with aging. So we're then looking at the systemic circulation. Then I'll show you data where we're now moving with our next grants into the gut and the role of the glycome and the microbiome with regards to HIV. Next slide. So what you can show is the levels of sialation of the anti-inflammatory glycan. So when you have the sialic acid residues as part of the glycome structure, those are anti-inflammatory. And what happens is those go down and are reduced during aging and also in ART-suppressed HIV infection. So you see both in the context of aging and in the context of HIV, you lose this sialation of your glycans. Next slide. We've then gone on to see further in our work One other point I might add, in HIV, you also have an increase in sialidase. So the actual enzyme that removes in the systemic circulation those sialic acid residues goes up in HIV, which might be the mechanism. And we're now actually thinking about interventional strategies. But we're also interested in the gut glycome and its regulation of the microbiome. So this is now a whole new field and an opportunity for students like yourselves on the call today to think about some new things to study with regard to the interactions of the microbiome. And as you can see then, there's a sustained opportunity to look at the environment and the equilibrium in disease. Next slide. What we're asking is that there are key glycan differences, as I mentioned, in sialation and fucosylation between the normal and the age-associated gut. In the normal gut, you have sialic acids attached to the glycans and high fucosylation. But in pathogenic states and age-associated gut, you have an increased sialidase activity, so you're losing the sialic acid molecules. You have an increased free sialic acids, improper fucosylation, and when you alter, then you get the gut barrier disruption and the systemic inflammatory markers are found. So you get microbial translocation and inflammation. This is affecting at the level of the sialation and glycome, not only on the gut epithelium, but also with regard to the microbiome. And so we're now looking at how sialic acid catabolism can alter the whole of the gut microbial composition, leading to, as it's stated on the left, microbial dysbiosis and gut inflammation. And again, there are several publications on this. Also, when you lack proper gut fucosylation, you can alter gut microbial composition and the same. So these are important new features we're now discovering, and we're studying these both in my lab in HIV and in aging. Next slide. Next. So what we've done is we've begun to study this in our HIV population, and we published this data about 2 years ago in mucosal immunology, where we were able to actually get gut samples from our HIV populations. So we sampled the right colon, the left colon, and the ileum. And in this case, and as was stated earlier, we actually were able to do RNA-seq, metabolomics, and microbiome analysis on all of these samples. So we're doing a full evaluation as well as glycomic profiling on this. So we have a full omics evaluation of all these parameters. I'll just show you some of the data today that we've generated. And this is a topic of a current R01 in my laboratory along with other collaborators. Next slide. So in this case, we're looking at RNA-seq data. This is now taking gut tissue and looking at mucosal RNA-seq. And what we were able to show then in our initial data is that there's a connection between gut hyposalation and a activation of an inflammasome mediator, ELF2. Can you click one more? And in this case, when we've done pathway analysis, as you've heard in the first speaker's talk, we were able to look at the genes and show that there's a correlation with ileum of the gut, the ileum biopsies we've done, with HIV DNA and inflammation associated with this change. So we're actually able to link changes in the gut glycome with changes in HIV directly at the tissue level here in the ileum, so that there is a direct modification. When you modify the gut glycome microbiome, you directly can affect the reservoir of the virus, HIV, now in the gut. And now we have a potential to look at this question of how do we go after that therapeutically, which I'll talk about in a moment. Next slide. Next. So we've also looked at pathway analysis in here with regard to changes in fucosylation as well. So the two targets, as I mentioned, is gut fucosylation and sialation. And both of these seem to be involved in this inflammasome pathway. So now through the pathway and single cell RNA-seq analysis, we're able to make this. So as you can see here, gut fucosylation is linked to the inhibition of the inflammasome-mediated ELF2 signaling, which is part of that. So now we have a target gene in ELF2, part of the inflammasome pathway. We can now think about altering and seeing what we can do then to the outcomes of gut permeability, ultimately leading to the non-communicable diseases I mentioned earlier. Next slide. So here's where we are today. We're thinking that host glycosylation machinery may hold the key to new biomarkers and therapeutics that could be harnessed to prevent the development of inflammation and age-associated diseases. And as you see below, we're looking at HIV causing hyposalivation and inflammation and HIV-associated comorbidities. We're now, and can you click one more? Okay. We're now looking at this further in other clinical outcomes. In this case, we're linking hyposalivation to neurologic impairment. So the changes that are occurring both at the gut level and systemically, we can show in HIV are associated with the CNS. So changes at the gut and systemic level and the glycome can lead to alterations in loss of the sialic acid residues and HIV-associated neurocognitive disease or HAND. We've also, on the next slide, shown that this is linked, hyposalivation is linked to the risk for cardiovascular disease. So not only do we have pathogenic pathways to study, but they're directly linked to these important non-communicable diseases or disease of aging, neurocognitive and cardiovascular disease. And what we're currently doing using monkey models of SIV infection, we're altering the sialic acid and fucose residues to see if we alter those directly in an SIV infected animal model, non-human primate model, can we now return back and alter these pathways for cardiovascular neurocognitive? And ultimately, we hope this will lead to novel therapeutics in HIV, but not just in HIV, as I've shown you, but these same pathways are critical in aging. And so we may have a new and novel approach with altering the sialic molecules, the host microbiome, and the outcomes of cardiovascular and neurocognitive diseases of aging. Next slide. So I mentioned to you, I will talk a few moments about SARS-CoV-2 and COVID-19, which is on everybody's mind. And as today we're talking about the microbiome, there has now been a strong link made between the gut-lung axis. And so that's the question, is there a potential link between lung inflammation and the gut during COVID-19? And you can see here that what we're now finding, as I'll show you some of our data, that there's an increase in gut permeability in COVID-19, and that may then lead to further lung damage. And on the left, you can see a number of the published papers around the gut-lung axis. This has become a very popular and important field of study. Next slide. So what we've done is we took a systems serology approach to identify correlates of COVID-19 severity and what happens both systemically and at the gut level. So here we're combining systemic markers of mucosal translocation, inflammation markers, metabolomic markers from our microbiome, lipidomic markers, and glycomic markers. And we've looked at this in acute COVID patients, both at the outpatient and inpatient level. Next slide. And here's just, again, some of the literature that says that the gut microbiota composition reflects disease severity and dysfunction in immune responses. And the gut microbiota alterations is important as well. So there, again, is a whole range of papers coming out on this whole important area. So those of you interested in microbiome, one of the most fruitful areas for study now can be in COVID-19 or SARS-CoV-2. And my lab has taken this up in a very strong way to look at it both in the acute with vaccine responses and then in the post-acute sequelae of COVID or long COVID in PASC. So I think, again, as students, a great area for study and investigation, and I'd be happy to talk to any of the students today on the call about potential collaborations. Next slide. So what we found is that COVID-19 severity is associated with higher markers of tight junction permeability. Again, COVID-19 affects the gut permeability, and the marker of interest is zonulin. This is a marker, it's a tight junctional permeability marker that's found to go up, as you can see on the left, in severe COVID and those with moderate COVID. And on the right-hand side, if you look at those individuals who are now deceased, they have even higher levels of zonulin. And on the right shows you the model of what the role of zonulin is. It's maintaining gut permeability, gut barrier function. So when you have higher levels in the blood, that means the gut is more permeable. There's also now the ability, because zonulin, there are drugs that can be used to target zonulin. And there's actually been a paper published in pediatric diseases with COVID, treating with these inhibitors of zonulin and maintaining gut barrier integrity. We're now taking the same approach to look at this in animal models of COVID-19 infection or SARS-CoV-2 with the Tulane Primate Center, and actually using these as therapeutics, potentially to modify the gut and eventually hopefully lead to and reduce some of the symptoms of COVID. And you can see some of the clinical work here that's referenced at the bottom of the slide. Next slide, please. We've also looked at other microbial markers, and I'll point to the one called beta-glucan, which is the third from the left. Beta-glucan is a fungal translocation marker. So we've heard about bacteria, we've heard about viruses. Now there's an important role for fungal translocation, especially in COVID-19. So we now think, because beta-glucan goes up at the highest level in the most severe disease, that it is about the microbiome or the fungal species present in the gut that are critical for altering the gut during COVID, and again, provides us novel targets for looking at this. So these are, again, all biomarkers you might want to incorporate in some of your own studies and think about from a therapeutic potential as we are. Next slide. We're also looking at plasma metabolomics. So again, linking what goes on from not just who's there and doing all the shotgun analysis you've heard about, or 16S, but now we're moving on to more metabolomics. And one of the critical pathways we find is the tryptophan pathway, which is another gut microbiome pathway in terms of producing kynurenine. And kynurenine is a pro-inflammatory molecule. So when you reduce tryptophan through an enzyme called indolamine 2,3 dioxygenase, and you'll hear more about that in a moment in our HIV studies, that also is present in COVID patients and leads to a hyperinflammatory state through potentially the gut microbiome. So again, by studying all these metabolomic markers, we can begin to understand more about disrupted gut function as well. And we have another whole field of study to incorporate. So the microbiome, the glycome, and now the metabolomics as well. Next slide. So where we are now with our COVID work, COVID-19 is associated with plasma markers of disrupted intestinal barrier integrity, higher mucosal translocation, and intestinal dysfunction. These results do not imply that mucosal is the primary trigger, as this is a complex, but this robust literature now indicating a disrupted intestinal barrier and microbial translocation may be important as another contributor. We're also finding in our post-COVID studies that there's a persistence. So this is, as you can see, putting fuel on the fire of what happens in the lung now can also now go to the gut. And there are studies that have been published in a number of cases finding the virus, SARS-CoV-2, in the gut tissue. So it does get to the gut and may be persistent and may contribute then to these sequelae of long COVID or the post-acute sequelae. So again, new targets to think about in our COVID-19 studies. Next slide. So I'll now move on to talk more about the gut microbiome and non-communicable diseases and HIV, as I've alluded to. Next slide. In this case, I mentioned to you the tryptophan pathway, and this is critical not only potentially in the COVID space, but also in HIV. So tryptophan catabolism is due to this enzyme 2,3-dioxygenase, or IDO pathway, breaks down tryptophan now to kynurenine. And this can occur both by a host enzyme and a bacterial enzyme that can contribute to this pro-inflammatory state. And if you look on the right, IDO activity correlates with loss of gut barrier promoting Th17 cells in SIV, and the abundance of gut resident lactobacillus correlates with low IDO activity in Th17. So again, HIV itself can disrupt the host microbiome, leading to alterations in this important metabolic pathway. Next slide. This has now been linked in our studies, and we've published this, to plasma tryptophan is an independent association with risk of carotid artery plaque. So these metabolic products, and this is human studies now in 1,000 HIV patients that we've published on, that when you alter the gut microbiome, reduce tryptophan and increase kynurenine, this is now linked to this important clinical outcome of cardiovascular disease. Next slide. And so the catabolism and related metabolites are altered. The plasma tryptophan kynurenine ratio now can be correlated with gut microbiome. So now we have an important systemic marker we can use in our evaluation. We can look at these ratios of metabolites and see what happens then related to the gut microbiome. And we're doing this much more shotgun metagenomics, as you heard about, in stool samples from large numbers of our HIV patients and correlating it back to these systemic metabolic changes. Next slide. So as I mentioned, there's an important role for microbiome-derived metabolites, and these just summarize a number of those, including the short-chain fatty acids, also succinate, and other secondary bioacids. So it's really now more about metabolomics in many of our microbiome studies than it is around just doing the shotgun analysis of the microbiome itself. Next slide. And we've now studied and showed associations of a variety of metabolites, including choline metabolites and carotid artery plaque. This was published in the Journal of Infectious Disease in 2018. Next slide. We've also looked at TMAO, and this is another important marker that's involved in a metabolic product of our microbiome, and it was significantly associated with plasma kynurenic acid, increased monocyte inflammation by soluble 14 and 163 that I showed you earlier, but not with HIV infection itself. So again, we have new pathways to study and new pathways to think about intervention. These same pathways have been looked at in aging and aging-related cardiovascular disease, so it gives us, again, a great opportunity to study this in detail, both in aging, HIV, and as I mentioned, in COVID patients. Next slide. So in the last part of my talk, I just want to discuss a bit about therapy. We've heard a bit before, how do we actually alter or modify the host microbiome? And what we want to do is look at several areas. We want to look at potentially prebiotics, which is the actual food for the microbiome or the bacteria. We want to think about probiotics, which is the bacteria themselves. We want to think about postbiotics, giving the short-chain fatty acids or other metabolic products. Then we want to think about combining these, which are known as symbiotics. And then finally, you heard about fecal transplants and some of the issues, but I'll show a little bit of data on fecal transplants and HIV. Next slide. So here we're looking at commensal bacteria that ferment insoluble dietary fibers to produce short-chain fatty acids. So again, that's the food we can give to our bacteria. Those are the prebiotics that we can give to get them to make important mediators. And the one that seems to be most important across almost every disease that we study are the short-chain fatty acids, which have anti-inflammatory properties. And these include butyrate, propionic, and acetate. Next slide. So what we've done here in this study is we look at the impact of therapy. So when we treat our HIV patients, what happens to their short chain fatty acids in the stool? And what you can see here in these 3 or 4 panels in the slide, it's maybe difficult to get the details, but what happens is at baseline, we have low levels of all the short chain fatty acids. And then after a few months of therapy, they're still the same. But after about 6 months of therapy, the short chain fatty acid levels begin to come back up. So simply by blocking the virus itself, we're able to alter now the production of these critical short chain fatty acids and effectively change the levels of gut permeability. I'm not going to show all the data on these studies, but we do change systemic markers of inflammation and gut permeability as well, just on heart alone. Next slide. We've also gone on to use fermentable products in vitro. So now this is taking and doing stool fermentation with dietary fibers, the prebiotics. And you can see a list of these here, such as FOS, gargum, chitin, glucan, potato starch. These are all prebiotics. And what we're now trying to do is take the stool from our HIV patients and incubate them, as you can see, pre-heart, post-heart, or a control. And we're now actually developing personalized type of therapy for our patient. We're now taking the patient's own stool, we're incubating them with a whole series of prebiotics and determining which of those induces the best short chain fatty acid metabolism. And you can see then the difference here in terms of how they induce differential levels of butyrate is the target that we're looking at. So we're now able to actually devise a precision medicine approach for therapy in our HIV patients by doing these kinds of in vitro studies. And ultimately, we hope we can expand this into other diseases as well. Next slide. So I just mentioned briefly, there have been fecal transplant studies in HIV patients. Again, these are most effective, if you've heard about, in C. diff in our first lecture. And they're now an open label FMT from Open Biome providing the material. So these are now phase 1 and phase 2 patients who've gotten, with HIV, who've gotten fecal transplants. Next slide. And so what's been shown in these initial studies, this was before all the other FDA hold came on. It was safe and well tolerated. You did get engraftment, but limited without antibiotics. And so there may be important ways of priming before you give back the microbiome transplants, and very low alpha diversity that may impact the degree of engraftment. And oral delivery may increase engraftment versus giving these through rectal type of enemas. So again, in the future, there are going to be many more studies going forward to think about this. So in my final slide, next slide, please. I'll just show you a little bit of targeting new therapies next. If you could just click one more. That we're now looking at this as a tree for targeting the systemic inflammation. And you can see the trunk of the tree where things like IL-1 beta blocking the inflammatory response. We have canakinumab and other trials we're doing in HIV and COVID. We're looking at JAK-STAT inhibitors like ruxolitinib, again, HIV and COVID. We're looking at IDO. We're actually targeting and blocking the IDO pathway. We're looking at metformin. Metformin is interesting. That's a drug used in diabetes, but it also can be used to alter the gut microbiome. And we're looking at that in aging, HIV, and COVID patients as well. And then finally, we're looking at other molecules like GLP-1 to affect the metabolic pathways. So we have a lot of studies ongoing. So my lab does studies both in primate models in collaboration with individuals in Tulane or Pittsburgh, and we do a lot of human trials as part of the AIDS clinical trial group program that was mentioned in my introduction to do all of these as therapeutic opportunities to study the interaction of the host and the microbiome. So I think there's a great deal of opportunities out there for students like yourselves to participate in bench to bedside to community type studies around the microbiome, HIV, aging, and even moving more into the COVID SARS-CoV-2 space. So I'll just, on the last slide, thank my collaborators here from the University of Colorado, UCSF, University of the West Indies. I also work with folks in the Caribbean, and I've worked with Puerto Rico and the University of Puerto Rico for almost 25 years with collaborators in HIV. I work with folks in Canada at McGill, University of Utah, and at Wistar Penn. And again, all of the grant support I've gotten from the NIH. I'd like to thank you for your attention, and I'd be happy to take any questions.
Okay, now we open the session for questions. Thank you, Dr. Landay. The first question we have here is from Dr. Godoy. Salivary sialic acid to Foucault's has been assessed for oral cancer patients. Have you looked at salivary samples for your HIV patients? Could it also be a mediator of the oral inflammation?
Great, yeah. We have not. That's a great question. It's probably something my collaborator for most of the glycomic studies is Dr. Abdul Monson at Penn. And what we've done so far, we've done mostly systemic studies with samples from the blood, and then we've done the gut studies that I've also alluded to. I think that's a great question and something we haven't done. And we are certainly collecting in many of our HIV studies oral samples, but we haven't gone back to do much of the glycomics analysis there. So I think that would be a great opportunity to do those studies and correlating it back with the oral microbiome. We have done a little bit of work there.
Okay. For our second question is from Dr. Gonzalez. Do you foresee a future lab panel of metabolites and markers as an assessment of inflammation and gut dysbiosis that can be used to guide interventions for treatment of chronic disease? At what time horizon?
Great question. I'd love to say soon, but we're still in that stage of identifying that. I think as we've heard, our analytic tools have gone just out of sight in terms of our abilities to look at multi-omic approaches. And I think that's where we want to be. We're now beginning to identify like TMAO, choline pathways, tryptophan pathways. We have a whole series of metabolites, and I absolutely believe that in the future, we'll be able to incorporate these, which we are looking at in larger aging studies now at the metabolites of coming up with panels. What's the time horizon? I think it's going to take some time to get these validated and into a point where then we can run these as routine markers for patients coming in. But it's really the wave of the future. I think gives great opportunities to students to start to investigate these questions.
Okay, we have a third question from Imlis Rodriguez. I'm curious to know if in the studies using plasma samples from HIV patients, these patients were on antiretroviral therapy. It has been shown that antiretroviral therapy can accelerate aging in HIV patients. If they were on therapy, how can we distinguish HIV effects on the microbiota from the effects of the drug in the microbiota?
Great questions. And that again is a good question because we've been studying the question of what effect does the drug have, what effect does the virus have. So we're now starting to look at in the patients who are getting PrEP, who are HIV negative, to see what is the direct effect of the drugs on the microbiome independent of the virus, because that's exactly what you need to do. And there are many new studies coming out asking that very question. With the newer drugs, they seem to be much less effect in the former drugs, like the integrase inhibitors, don't seem to have as much negative effects as we may have seen for some of the what we call D drugs on metabolism or mitochondrial function. So the current drugs are very different than those that were used even just 5 years ago. So I think we now have a greater armamentarium of drugs to treat with, and then we'll also be able to study those aspects in our PrEP patients who only get the drugs and aren't yet HIV infected.
Okay, I think we have time for one last question. The last question is, have you considered the archaeal community in gut microbiome study?
I'm sorry, what community?
Archaeal.
Archaeal communities, yeah. Again, a great question. We haven't looked at that, but that's certainly something to be considered in the future. I haven't seen much done on that where, most of the work, as we talk about, is on bacterial. There's more virome and now microbiome. I think next should come the archaea as the next sort of phase of study to see as we look at all the microorganisms, because the microbiome is all of those together, not just the bacteria, which is where most of the studies have been to date.
Okay. Well, I thank you again, Dr. Alan Landay, for your time and for that excellent presentation. I hope everyone enjoyed this interesting presentation. And to access the next session, click on the open door icon at the bottom left of the screen of the current sessions. And when you see the agenda, click on preview icon that appears to the right of the session title to access the session. If the session has not yet started, a timer and background music will appear until the moderator and the speaker or production are in the session.
Thank you again. Thanks, everybody. And happy to follow up with any individual questions or experiments people are interested in. So please email me.
Thank you, Doctor.
screen that I'm seeing right now is from the computer or from the lens? Allow you to use the same screen. beard. Don't worry about that. Just go for the presentation. Yeah, we'll give you the question. Let me know when you are ready. You ready, Lorraine? Okay, let's do it. 1 second. 1, 2, and 3.
attendees will interested in asking questions and operating next to the speaker of the session will need to press the grab the mic icon at the bottom left of the screen. The production will grant access on first come, first served basis. Grab the mic won't be granted by production while the speaker is giving her talk. During the Q&A session, questions can also be written in the chat, and the 5 minutes for Q&A will apply to both questions live or written in the chat. Once the session is over, and for attendees to access the following session, we'll need to click on the open door icon at the bottom left of the screen of the current session, and then you will click on the preview icon that appears to the right of the session title to access the session. If the session has not yet started, a timer and a background music will appear until the moderator and speaker or production are in the session. And on one last final disclaimer, the content of the session and all other sessions cannot be reproduced without prior authorization from the organizer, the Department of Microbiology, UPR Medical Sciences Campus, and the production Vistazo a la Ciencia. So now I will be presenting the speaker. My name is Lorraine Nicole Velez Torres. I'm a PhD candidate in microbiology at the UPR Medical Sciences Campus, and today I have the pleasure and honor to present Dr. Kerry Kinney. Dr. Kinney is the Gilvan Centennial Professor in the Department of Civil, Architectural, and Environmental Engineering at the University of Texas at Austin, as well as a professor in the Department of Population Health and the Dell Medical School. Her cross-disciplinary research in environmental engineering centers on the investigation of human exposure to contaminants in the built environment. Her research team investigates microbial communities, pathogens, allergens, and pollutants present in a range of settings, including homes, schools, and commercial buildings. Her current research focuses on microbiome and pathogen studies, bioaerosols, contaminant exposure pathways, as well as identifying the relationship between indoor exposures and the physical and mental health of building occupants. Dr. Kinney is currently working on a large interdisciplinary study led by the University of Puerto Rico to examine the relationship between home water damage, respiratory health, indoor fungal communities, and inflammatory potential of particulate matter for residents in Puerto Rico following Hurricane Maria. Over the last several years, Dr. Kinney's research group has also investigated the particulate-bound contaminants present in low-income homes of minority children with asthma in the Central Texas region. These studies have led to a greater understanding of the conditions that promote fungal growth in buildings, new indoor sampling strategies like filter forensics, and insights into the relationship between environmental exposures and asthma outcomes. She has over 160 publications throughout her career with research support from NIH, NSF, EPA, and the Sloan Foundation. She's currently the co-chair of Whole Communities, Whole Health, a Bridging Barriers research program at UT Austin, and she's also the president of the Society of Indoor Air Quality and Climate, or ISIAQ, Healthy Buildings of America Conference 2021. And this year, she was also elected to Academy of Fellows from the same society. I've had the opportunity of working with Dr. Kinney, and I have learned a lot from her. She's definitely one of the researchers that I admire. She also has a great sense of humor. So without further ado, let's welcome Dr. Kerry Kinney that will be talking today about the indoor microbiota of the built environment. Welcome, Dr. Kinney.
Thank you. And thanks so much, Lorraine. I figured if you kept talking, I wouldn't have to do a talk. It'd be much more fun. So thank you for the invitation to speak today. I've actually been taking quite a few notes for the previous two speakers. I'm a little bit different among this group, to be honest. I'm an environmental engineer who I started in biological treatment, became interested in human exposure to both microorganisms and other pollutants in indoor environment. And in fact, today what I'm going to talk about is indoor microbiota in the built environment. And I'm going to give you a little bit of a tour through how an engineer thinks. And don't worry, it won't be that horrible of an experience. But just as you guys are looking at, looking at the kind of the human body, we're looking at the built environment and trying to understand what are the microenvironments for different microorganisms, what happens when things go wrong, and what are the implications of that with respect to human exposure to potential pathogens or microbial communities in general, and how can we essentially design a healthier built environment. So what drew me to work, kind of this transition, if you will, from basically using microorganisms as little mini engines to treat contaminants in the environment, which by the way I still do, I became very interested in exposures. And when you think about where we get most of our exposures, it really has to do with the indoor environments. Americans spend almost 90% of our time indoors, and there's quite a bit of effort looking at outdoor exposures to air pollutants and other even potential pathogens. But really, if you're spending nearly 90% of your time indoors, and for us that means in environments that we've constructed, what can we figure out, how does the built environment essentially affect our exposure? And really, outdoor exposures are not necessarily equal to indoor exposures. For a while, there was only this focus on outdoor, and now there's really this growing awareness of how important the indoor environments are. And so pollutant concentrations, and along with that, I would say the microbial composition that you'll find in indoor environments are very much a function of the building factors and, frankly, occupant behavior. So whether you're running your HVAC system frequently or you open the windows and get natural ventilation, that will affect, of course, what penetrates into the indoor environment. And also there are a variety of exposure pathways that are possible. So we often think about, the airborne route, but for instance, if you look at the little icon here in the lower right-hand corner, we have drinking water systems that are also very rich in, we have a rich variety of microbial communities present. And that also, we purposely plummet to our homes, and that also can have an effect on the indoor environment. So what we've looked at is doing a couple of approaches when we do these indoor exposure assessments. So we've done quite a bit of field work where we go out into actual buildings of many different types and collect samples, airborne, swab. You can use very sophisticated, high throughput air samplers, or you can do relatively low tech but effective settled dust samplers. And as I'll talk about later today, you can also exploit what's naturally present in a lot of, at least mechanically ventilated buildings, and actually sample home filters. So we've done quite a bit of work in actual buildings. And with that, you realize there's actually quite a bit of complexity that is really not particularly well understood. So I'm going to talk about that in a moment. We also use what I call simulated environments. And so, for instance, at the University of Texas, we have a full-scale test house. And it's only simulated in that they, for some reason, when we got that house, they wouldn't let us all move in. So it's an unoccupied test house. And as a result of that, we get to adjust all the different factors. We can run water in the system. We can burn our fajitas in there and see what kind of pollutants are generated. And we can adjust and control the ventilation very carefully. We can release aerosols and track them through the environment. So we learned something from both of these. In the actual buildings, you learn about the real complexity, even in something relatively simple. People think in terms of ventilation, it can be quite complex. And then within these simulated environments, we can go from chambers to full test house to control certain factors and explore them in, specifically, what does natural ventilation do, for instance, to indoor exposures. We also look at what I call common exposures. What's the everyday exposures you and I are getting? And then, as Lorraine mentioned, we also look at what I call extraordinary events, which is a nice way of saying disasters. Okay, so whether you're talking hurricanes or you're talking the COVID-19 pandemic, we can take many of the tools that we use for kind of everyday exposures and use those to understand how these natural and pandemic disasters, if you will, are affected, how the built environment and how we've designed it are affecting essentially the outcomes of these events. We've also been trying to figure out how we can partner with communities for data collection. So one of the challenges, of course, is that, and I don't need to tell all the folks here, but when you want to collect samples from many, many buildings, and for instance, homes, and working with vulnerable communities in particular, can we actually partner with them and actually have them send us the samples? And can we also leverage technology to more efficiently assess exposures, occupant behaviors, how do you clean your house, et cetera? And, what are the conditions within your building? Do you have, I'm as interested in whether you have carpet on the floor or hardwood floors as I am in how you run your HVAC system, your heating, ventilation, and air conditioning system in your home. So can we leverage technology? Specifically, we're working with Whole Communities, Whole Health, for instance, to develop what we call the Horn Sense app, sorry for the Texas theme there, to basically not only use that for communities to report issues and to provide data to the team, but we also are going to use it as a dashboard to provide data back to our community members. So I'm going to do a little walk through Building Lane, and and this is just a snapshot, if you will, of the buildings that you will see in the central or in the Texas region. And on the left-hand side, you'll see two types of classrooms. One where there's what we call the permanent buildings in the top left corner there. And you'll see that's a brand new or relatively new building. It will have high efficiency, likely high efficiency filters in the ventilation system. We'll have many classrooms. And then even at these relatively nice buildings, we would say, new buildings, modern buildings, you will also see what you have on the left-hand corner there in the bottom left-hand corner. And that's what we call a portable classroom. Okay, so that's also being used at classrooms all the way from K through 12. Okay, the students might spend part of their day in that nice building and the rest of their time in that portable building. It's also called a temporary building, and essentially it's neither of those. In our area, over half of these are 25 years old, and they're just shipped from one school to another as they use them to expand and deal with changes in population. So buildings certainly vary. So those are just two school buildings. And then if you look on the right-hand side, these are just a couple of homes in our area. So a standalone home. You can also have essentially, again, these are called mobile homes or trailers. Generally, they're not moved very often. They're just, affordable homes. And then, to be honest, there can be many different types here. You can have dorms, you can have apartments, but you get the idea. There's a wide range of buildings, and how you ventilate those buildings, how it affects the water supply coming into the building, et cetera, varies depending on where you are in the country, what type of ventilation is being used. So specifically, these are looking at, we're in a very hot and humid environment, and we have a very mechanical ventilation is in almost every building, but certainly in our project that Lorraine mentioned, natural ventilation is very common as well. So within a building, then we often think about, okay, you're just looking at the exterior of the building and thinking, okay, I can figure out everything I need to know about this building just by looking at the outside. But you also have to think about all the systems that are, I'll just use the word plumbed into these, right? So there's the ventilation systems that I'm going to talk about today. And if there was any, and there is no bright side, but if there's any outcome from the COVID-19 pandemic that could, potentially be useful going forward, that would be that I think the general public and building managers everywhere understand the importance of ventilation now. But also, you certainly have drinking water systems. They're being used of course, for drinking water, but they're also being for your daily, used in your daily shower. And you can see there in the lower left, that's the biofilm that builds up in a shower head over time. And then certainly we have, of course, the wastewater system and the plumbing associated with that. So I wanted to start with how engineers view buildings, okay? And then I was going to kind of walk through, the microbiome and how these influence the microbiome that we recover in buildings. And so this is what we, we like our arrows, we like our mass balances. And so if this is an indoor environment, very much simplified here with a single box representation, at the center, you see the indoor sources or emissions that are contributing to either air pollutants, or it can also be microbial emissions directly from occupants themselves. Also, both particulate matter and microorganisms associated with that particulate matter can be deposited onto floors and resuspended off of floors. These microorganisms, when they're in the air, can decay over time. And kind of overriding all of this is we have, of course, ventilation, otherwise known as air exchange, where you have, and I'll just put in quotations, fresh outdoor air being provided to the home. And the rate at which that outdoor air is provided to a certain volume of space is what we call the fresh air or the air changes per hour, the air exchange rate. Okay. You also have outdoor sources. And so, if you're looking at something like particulate matter, and I'll show you some data in a moment, really fine particulate matter, the smaller fraction, can actually penetrate pretty readily into indoor environments. And that can have an effect, of course, on the composition of both the, for instance, if you're looking at biological, like the microbiome, you have ready penetration of fungi, for instance, from the outdoor to the indoor environment. And then there's other, kind of mitigation measures beyond the decay. And so I've shown in the top there what I've labeled the ventilation system. You probably know, but I'm going to show you plenty filters. So now you can go change your home filter, is that there are filters that are built into both home and, school ventilation systems. What's interesting about these filters is they were not originally designed to actually protect and reduce pollutants from the air, although they certainly do that. They were really used to protect the downstream equipment in the ventilation units, in the HVAC units. Nowadays, of course, we also have the possibility of installing these portable air cleaners, and I've just shown an example there. And so you have a certain volumetric flow rate of air going through that filter, Q sub C, and then it has a certain removal efficiency. And really what you're looking for, if you're looking at comparing different air cleaning systems, is what we call the clean air delivery rate, which is essentially the product of the control efficiency going through that device, which, a lot of times systems will report very high removal efficiencies. But if they don't move any air, if they're not treating any air, in other words, if Q isn't very high, your volumetric flow rate through the system, you've actually done very little good. So actually the product of the two is really the value you're looking for. So when engineers look at the built environment, we're looking at all these different systems. And in fact, this is a highly simplified model of some of the processes that go on in the indoor environment. But what I'd like to do is kind of walk through a couple of these today and how they influence what we see in the indoor microbiome. I also would be remiss if, as an engineer, we didn't actually present one mass balance model. And so if C represents the airborne pollutant, or it could be the microbial concentration present in the indoor air, you can look at the change of that concentration as a function of time. dC/dt is equal to essentially what pollutants are penetrating from the outdoors minus essentially a series of removal terms. And this is when you're looking at like kind of the infectious risk modeling or the indoor modeling. Essentially, they're adaptations of this very kind of fundamental model. And so if you think about some of the ways that you can remove microorganisms, or frankly, they've been adapted for quanta, removing SARS-CoV-2 from the indoor environment. You can look at ventilation, and lambda represents the air changes per hour, which is simply the volumetric flow rate of outdoor air going through that space divided by the volume of that space. You can also look at the effect of an air cleaner. So remember, there's the clean air delivery rate. You can look at other loss mechanisms, which either could be deposition onto surfaces, because again, we're looking at the airborne concentration, or decay and inactivation if you're thinking of microbes. And then ultimately, the final term there, of course, is really important, is the emission sources. So what is driving the emissions of the pollutants into the indoor environment? And I look at microbial emissions, as well as, for instance, looking at particulate matter concentrations and other pollutants in indoor environments. And I just wanted to note or reiterate that, if we look at the steady state, if we ignore outdoor penetration and we look at the steady state solution of this, what you get is essentially the same airborne quanta infectious risk modeling results that you'll see more on, for instance, during the COVID-19 pandemic. They're exactly the same, but engineers have one term, and folks who are looking at risk modeling and other things have a second. They work with quanta. C is essentially quanta in this case. And so one of the things I'd like to encourage is for people that are essentially designing, operating, cleaning our indoor environments, the built environments, that we have a common language so we can talk with people who do exposure assessments and ultimately look at health outcomes, whether we're looking at COVID-19 risk of infections in indoor environments, or if we're looking at potential pathogens in the indoor environment, microbial, bacterial, fungal, for instance. So let's start with just one of those terms in those equations. Remember, the last term was emissions, and turns out that as we spend a majority of our time in the indoor environment, we are just a walking, shedding machine, if you will. So there's a huge imprint, or a very significant imprint of, and this is just looking, I think, at a sample of like 25 samples from the indoor environment, a very relatively small sample for us, looking at some of the kind of dominant species, and the ones you see underlined are, human associated. And of course, we're shedding skin cells all the time, and we have the skin microbiome that is certainly seeding the indoor environment. So as much as engineers like to ignore occupants, and those in the health field focus on people and occupants, there's this crossover. Okay, so occupants, people, occupy buildings, and we seed those buildings. But you can't ignore the outdoor influence. And I want to talk about some work that we've been doing with these, I keep using the portable, temporary, neither buildings, these basically rather shoddily built classrooms. And just to give you an idea, we estimate it's about 400,000 of these classrooms around the US. They're used very commonly as school budgets have shrunk. Okay. And so one of the things that we found rather interesting is that in some ways, if you look at the left-hand side, so that's a portable classroom, and then you look at the right-hand side over there, and that's that nicer, newer building. We actually, I think, sampled in both of these. You will see that, you look at that, the one on the left-hand side, and you're kind of expecting, well, maybe that's not going to get a lot of air. That's not going to have a lot of outdoor ventilation. And in fact, if you look at the figures on the bottom here, permanent classrooms being the more, kind of your traditional classroom buildings, and then these portables, ACH is air changes per hour. So the higher the air changes per hour, the greater the ventilation rate you're getting in a particular space. And what's interesting here is that we were quite surprised to see that the portable classrooms had actually much greater air changes per hour, which means there was more outdoor air penetrating into the space. And of course, there was a difference between day and night, and you need to keep that in mind when people are reporting their ventilation rates. If they're a building, for instance, that's only occupied during the day, they will often, in hot and humid environments, turn down that ventilation at night so that you're not wasting energy. And, you know, as we look to mitigate climate change, get to net zero in terms of greenhouse gas emissions, you're going to see more of this. So you have to also keep in mind that what's often reported for a building, you're thinking this is pretty good, but maybe you shouldn't go in at night if they're actually turning down the ventilation system as shown here. Now, I have to say this was pre-COVID-19 pandemic, and I have seen that there have been schools and universities, et cetera, that have decided to continue to run the ventilation system during the night as well to maximize, you know, essentially the flush out of anything in the building, specifically SARS-CoV-2 at all times. But anyhow, so one of the things, the first surprise was, of course, that the ventilation rate in the portables was much higher. And what we found when you looked at the indoor microbiome samples, we found a much higher fraction of outdoor associated fungi actually in the portable classrooms. And then in the permanent buildings, which tended to have lower outdoor air exchanges, although there was a great range here, tended to have a higher fraction of human-associated taxa. And what was happening? Well, when you actually started to look at these portables, and I'll show you some pictures in a moment that kind of make the point, we found, and we did a work with the building engineers who looked at different, you know, basically blower door tests and other things. They determined that there were a lot of uncontrolled airflows into portable buildings, and specifically what I call the hidden unmaintained spaces. So you were getting, depending on the weather conditions that day, et cetera, you would get uncontrolled air, for instance, coming from the attic. You might have the air basically from the attic, the unmaintained attic, I might add, into the classroom space, or it might actually be coming from the crawl space, depending on the pressurization conditions and the weather conditions that day. And then, of course, these systems, these buildings are not particularly robust. So you also have leaks through the building envelope, which is a fancy way of saying the walls. And I wanted to show you what actual looks like in some of these systems, right? And so these are some pictures for, you know, why did I say uncontrolled? This is why. So we peaked above the false ceiling in some of the portable classrooms, and you see that it is open to the outdoor air. It's a vented attic. In some cases, they had decritterified it, if you want to say the word. They'd had to multiple times remove critters, if you will, bats and other things from some of these spaces. Also, the picture on the right shows the what we call the duct tape solution, which means these they're basically wall air conditioning units, and they don't necessarily provide enough cooling load for the kids in the classroom. So you can close, this is taping off the outdoor air vent into the building. So you're going supposedly into full recirculation of indoor air, and that reduces how much outdoor air you have to cool. So that's an attempt to solve that problem. And then you can see, I told you that there's filters in all of these HVAC systems. That is true. In the lower left corner, you see that, yes, that's there, but in principle, but it's not very well fitting. And in fact, that wouldn't do you any good at all. And so one of the other things that's, you know, we were given access to work with some of these portable classrooms, obviously unoccupied. You can also look at what we call positive versus negative pressurization. And essentially what that means is if you positively pressurize a space, right, you can think of it as, you know, very simplistic way of blowing it up like a little bit of a balloon, right? So you're positively pressurizing the indoor space with outdoor air such that anything's going to leak out. You can have, unfortunately, negative pressurization, which we found it swung back and forth in these buildings. And under those conditions, within the space of the classroom where the kids would be, for instance, you can, we did some microbial source tracking, and you can see the microbiome from the attic, the imprint of that penetrating into the classroom. Okay, so why dig into the details of some of these buildings? Because the details matter, and also they affect, where are these microorganisms coming from? And once you have that, for instance, if I want to develop a mitigation strategy, for instance, I would work with upgrading the HVAC units, so they provide these exterior wall units, so they provide enough cooling load, and I would seal up, obviously, the attics there so that we could have controlled ventilation of the space rather than uncontrolled. So that's just, and this is more of an extreme example, but it does tell you what can actually happen in the field. So when you're thinking about, what microbial exposures people are getting in their everyday life, well, when they're in the permanent classrooms, they're getting one set of exposures, and then when they're here, they're getting probably another set. So I also want to talk about some more about these outdoor contributions. So this is just looking, similar to what I showed you before for the bacteria. This is looking at some fungal species, again, the same set of samples that I showed you earlier in the similar plot. And for instance, in the Central Texas area where these were taken, alternaria species are incredibly common and very robust, if you will. And they're also known to aggravate asthma symptoms for those, particularly for children with asthma, and frankly for me as well. So these are some of the outdoor contributions that you'll see. And then when we went to, now this is looking at the data in that, and we did it across 8 or 10 schools, looking at the microbiome that actually is present in the permanent classrooms now. So this is not looking at the portables at the moment. And we had 3 different kind of sampling locations. And so the first one, these are in the top left here, you see the desktop. In the center here is the door frame. And then in the right-hand panel in both of these is what we call dust collectors. And this is where, for instance, in real schools and other things where the teachers are talking, and this is, by the way, these samples were collected pre-COVID, you can't have a fan or a sampler going all the time because the noise is a problem for the teachers. So essentially, dust collectors are, we call them Petri teas, but you can take essentially Petri dishes, suspend them at different locations within the classroom, and they essentially serve and collect. In this case, I think we did a 4 or 5 day period. You can collect the suspended dust in the classrooms while they're occupied over approximately a week period. And so what you can see here on the left-hand side is, first of all, if you've ever wondered if desktops are dirty, they are indeed. So you see the human-associated bacteria are really dominating on the desktops, which just reflects that the students, of course, have are leaning on them, and in some cases, maybe spitting on them, etc. And what's interesting to me is that, and this is again, when you go into the real classrooms, you're like, oh, we're going to find a lot of dust on the floor. No, the floors were cleaned. And pre-COVID, the desks were not cleaned. So you see that, that result here. But what's also interesting is even on the, just looking at the top of the door frame, which is kind of a long-term reservoir, sampling location, and then these suspended dust collectors, you also do see a pretty significant imprint of human-associated bacteria. If you then look at soil-associated bacteria, notice the shift in the scale there on the y-axis for all of these. You have some folks who maybe rolled in the mud on the desktop there and brought some back in. But in general, you're seeing a lower abundance, and you're seeing it more evenly distributed, in the door frame, in the suspended dust collector, and to a lesser extent, probably due to the dominance of the human-associated taxa of the soil-associated taxa on the desks. But bottom line, what you're seeing is both an indoor, in other words, occupant and driven effect, and really soil associated outdoor imprint as well, but it matters where you sample and how you sample. And if you think about how do these, if you will, particles, bioaerosols, microbes get everywhere? This shows you actually real-time data. This is, I think, for only a couple days worth of data, that in the same set of schools, and this is just for one classroom. And what you're seeing here is particulate matter concentration. They have it as PM10, so 10 microns or less. That's kind of the bigger cut fraction. And the dark traces on the bottom graph here are the indoor measurements, and the light gray are the outdoor measurements. And first of all, you'll see that, well, this thought that these bigger particles, PM10, don't penetrate into the indoor environment is pretty good till about 8 o'clock in the morning. And then what happens is you have all these spikes. And if you look in the upper left corner there, that kind of blow up of a time between 7 and about 5 or 9 and about 5 o'clock, you'll see that these spikes, declines, spikes, declines, because what happens is these were high schools, right? The students shuffle in, class goes on, the students shuffle out, etc. So you get these, and then the ventilation kicks in and it's going to be dropping it. But what this is showing basically is you resuspend particles and microbes off of the floors just to do, occupant, in this case, student activity. So when you're thinking about loss terms, they're not permanently lost. If there's something is settled onto a surface, and especially if it attaches to a particle, it can readily be resuspended just from essentially walking and occupants coming in and out of a space. And I mentioned at the beginning that I care whether you have what we call laminate, that's kind of that tile-like flooring, or carpet. And the same thing, I care in your homes, whether you have hardwoods, a wood or a hard flooring, or whether you have carpet. And this just shows you summed across each of these for each of the size ranges. So the PM1 is the smallest particulate matter, less than a micron in size. This is the blue here is the 2.5 microns, so that's kind of the respirable fraction. Actually, these two are, and then PM10 are these bigger particles. And everything in clear, we're just looking at the concentrations in each of these bins, if you will. In every case, whether you're talking about PM1, 2.5, or PM10, you have higher concentrations if you have carpet. And fundamentally, carpet over time collects dust, and it's very difficult to remove it. And these are actually, and this isn't like thick shag carpet. This is kind of that carpet you see, that industrial carpet you see in schools, for instance. So even though they're being cleaned very frequently, you just can't get them as clean as the laminate. So that just says, not only do the building, like, how you run your HVAC system, how it's been set up, etc., matter with respect to where your potential exposures can come from, but like just even what's in that space with regard to materials, and specifically in this case, flooring. And this, we actually did this study before COVID-19, but I've been thinking about it a lot since the pandemic. So this is actually a top view of our full-scale test house. And it's a 2-bedroom house with 2 bathrooms. And you'll notice, because we're engineers, we have what we call the control room, which is where we put in all of our equipment. And we did it's nothing simple, but we used fluorescent particles, like 3 microns in size. We did a 15-minute aerosol release. And we actually turned the mechanical ventilation off in the system, and we only had basically the ability for outdoor air to come in, and then there was one window that was open an inch or two. Okay. And we had distributed throughout the home here sampling slides to be able to collect the aerosols and to figure out where did they end up. Okay, so the big blue arrow on the right-hand side here in this control room is where we did this 15-minute injection. We then waited, like the next day we came back and said, well, where did all these fluorescent, these labeled particles end up? And they ended up throughout the house. You'll see the size of the blue dot basically represents roughly the concentration we recovered in these different locations. I think the bigger surprises for me, though, is that you see there's a master bedroom closet, MRC. We found it in the closets. So if you're wondering why your clothes in your closet get dusty over time, you wonder no more. So even though you close a door, right, you have the gap below the door. So this idea that, oh, I've closed the door and therefore I've not allowed aerosols, bioaerosols, for instance, if you're thinking about SARS-CoV-2, to penetrate into these different spaces, you're unfortunately wrong. And I think the two red dots there are the ones that really did surprise me the most. Is those are basically within drawers that were closed within a closed cabinet. We actually also, and we have controls to show that it came from this injection, we actually recovered to a much lower degree, 1% to 2%. Okay, so not very much, but it penetrated throughout this building very readily. And then I wanted to comment on ventilation systems. So, and in fact, the rest of my talk's going to kind of focus on, particularly on what we've done with regard to looking as filters as indoor samples for the microbiome. So we're going to mostly talk about these ventilation systems that we have in our home units, which is illustrated here on the left. I also want to mention, though, when you go into larger buildings, whether it's universities, office buildings, retail units, retail stores, etc., right? All other kind of commercial or larger buildings, you're going to have what we call these centralized air handling units here. And these also have filters in them. So you can also sample them, sample those filters to get an indication of what essentially the airborne and what we look at as airborne microbial community that's present in the indoor environment. But I also wanted to comment on if you look at some, these like university buildings or commercial buildings, et cetera, where we've been trying to increase the fraction of outdoor air, you have to keep in mind that you're actually in these systems, you recirculate, although you run it through the filter again, you recirculate maybe 60 or 70% of the air. So for every 100 cubic feet or whatever that are going in that are supplied to the space, only 30 of those 30% is actually coming from outdoors. And with COVID-19, they've been trying to open that up, but you have a limitation in hot and humid environments because simply your air handling unit can't handle it, and you'll have moisture driving into your building that's uncontrolled, and you will have fungal problems as well. So I just wanted to comment briefly on that. So what we've been doing, and let's go back to that kind of home environment, it turns out we all have these filters in our heating, ventilation, and air conditioning systems, these HVAC systems, and they actually serve as long-term airborne contaminant and in case of what we've been looking at, microbial and pathogen samplers in homes. So they're widely available. And I will say this, of course, in mechanically ventilated space. Of course, when we were working with the team in Puerto Rico, we were working with mostly naturally ventilated spaces. So that gives you a completely different answer with, how you sample that. But in our, in Central Texas, well, all of the South, right? It's a hot, humid environment. We have these everywhere. So they're easily collected and they provide an integrated sample of the air within that indoor environment. And actually people have no trouble or no qualms about sending you their sample. So if we look then at these home filters, you will see that there are quite a few, and this was looking at 3 different sampling strategies, and we have settled on vacuuming the dust up. Human skin-associated taxa are well represented. Unfortunately, stool-associated taxa are also very well represented here, which isn't too surprising, but keep in mind, look at the access here. So it's only like 5%. It's not the worst thing in the world, okay? We also looked at a sample of homes in a rural community, and we had divided the homes into those with asthma. We'd sampled 40 homes with asthma, 20 without asthma. And we saw an interesting thing is that in the homes without asthma, they had higher concentrations of selected bacteria, and these were selected based off the Lynch study that found kind of the protective microbiome. We also saw it in this, sample of homes in a rural environment, but actually more associated with whether, they had no asthma or either intermittent or persistent asthma. So there is there is kind of a protective effect for microorganisms. So not just the point that an earlier speaker made, not all microorganisms are bad. And in fact, we often want rich microbial exposures. It's just the pathogens that we're most concerned about. We also looked at this is again this filter forensics study where we're looking at what's built in the filter here. We were looking at the indoor fungal community structure, and we were taking quite a bit of data, collecting it from our participants to see what was driving the structure of the indoor fungal community that we recovered from these 60 homes. So we saw a strong seasonal effect, which was expected, and others have shown that as well. We also saw relationships to the how close this was a semi-rural area, so how close they were to active ag fields, whether they had carpet, so you'll see that keep coming up, how much they ran their air conditioner. We also saw some relationships with phthalates and flame retardants and the structure of the fungal communities that we recovered off of these filters, and also with the severity of asthma as reported by the participants. And I just wanted to briefly mention, because everybody talks about the forever chemicals, guess what? The forever chemicals are very readily collected in your home filter dust. And in fact, in this first study, we looked at phthalates and flame retardants and found, in general, higher concentrations within the filter dust than is present in the settled dust. And keep in mind, anything that gets captured in your filter came from the air within your home, so it is actually an air sampler in a sense. And then I did just want to mention, because the COVID-19 pandemic continues, you can take also these techniques and use them to look for the RNA signal for SARS-CoV-2. And this is just showing, the results from a single home with 2 COVID-19 positive occupants. This was actually 2 months after the onset of symptoms, and a month after the resolution of the symptoms. And if you just look at anything in red is where we recovered the signal. And specifically, if we're looking at particle-bound SARS-CoV-2, and this is in copies per gram, we recovered it from the home HVAC filter unit, whether it was via swab or vacuuming the dust up, and at pretty high signal strengths. And this was actually a pretty darn low efficiency filter that was in place in that unit, and it still recovered quite a substantial signal. We also saw that any surfaces that were routinely cleaned actually had greatly reduced signals. And just to give you an idea of what we were seeing, just looking at N2 copies per gram of dust, if the floor in the master bedroom where the occupants were staying, it was a fairly high level still of the SARS-CoV-2 RNA signal, the N2 gene. And then in the HVAC dust, you also see a pretty strong signal. And then as a negative control, we also evaluated HVAC dust that had been collected pre-COVID-19. So we're continuing to use this methodology to look at multi-story buildings and others where essentially we have these centralized air handling units as basically a surveillance method. The next thing is, and just to briefly touch on this, is that other sources that you don't necessarily think about. I think people now think about occupants, and now you're going to wonder about your flooring, but also, and the outdoor contributions, also how we run our plumbing systems, right? And in fact, how it's treated at the drinking water plant affects one of our daily exposures to water, obviously, is from our shower. And this is showing on the left-hand side, we had a low flow and a high flow shower. And this is just doing a visualization using basically a high-speed camera with my collaborators at Texas A&M. And bottom line, we show that showers generate very small droplets that can be inhaled deeply into the lung. They're an important microbial bath, if you will, that we get every day. And we've also looked at what's found in drinking water, right? So the drinking water, they use what's running in through your tap. And this is just a study that we've been doing pre and post, or I shouldn't say pre, throughout the COVID pandemic. And we are sampling drinking water at an academic locations, commercial locations, some additional residential locations. And you can see that these are looking at the bacterial communities. Even at the phyla level, you can see these pretty strong variations. And for instance, you again see a seasonal effect for that impact. So in the interest of time, I think what I'm going to do is just skip to one other element that I'd like to talk about, which is how can we collect data remotely? So rather than we go in and collect the samples, we actually have asked our participants to collect the dust samples off of the home filters. And what we see, for instance, here on the left-hand side is that the participant gathered samples, they actually do a pretty good job. There's a bit of greater range in the mass that they collect relative to researcher collected samples. And we do see some differences in the microbial community, but mainly if you don't collect enough dust on the filter, if it's a really low sample size. Okay. And finally, I just wanted to, in the interest of time, just get to the closing thoughts. The previous slide is just showing some of the work that JP Maestra and others have done looking at mapping the UT biome. These are working with students to collect samples from across the campus. But in closing then, I just wanted to reiterate that both, if you're looking at indoor microbial assessments or pollutant exposure assessments, they benefit from understanding the fundamental processes that are occurring in buildings, kind of this ideal ventilation and actually what you see. Outdoor does not necessarily mean indoor when we talk about exposures, but we need to understand both. We've been implementing this remote data collection via, smartphones and participant supplied samples, and that is possible. And one way that we can extend the reach of these studies. This filter forensics idea is certainly useful for looking at the indoor microbiome in mechanically ventilated spaces. And then I think the future is combining all these different types of data collection modalities to capture kind of richer information about the occupants and their activities, and then certainly the exposures that occur within those. And with that, these are the acknowledgements for our funding, and I'd be happy to answer any questions if there's time.
Thank you so much, Dr. Kinney, for an excellent presentation. We do have some questions. We have one question from Marco Cardi. Have you used some software for tracking shared microbial taxa? If yes, what kind?
Yes, so we've done, so it's very, one of the things that we've done is use these microbial source tracking algorithms that were originally actually developed to see if there was contamination of, in the high throughput sequencing that people were seeing in some of the early work. We actually use that to track, how much of the outdoor microbiome, for instance, is penetrating into the indoor space. Or if we see, for instance, the attics, we take samples of the dust in the attics and then we change the pressurization. And that's what we do to, examine, figure out where the microbes are coming from, right? Because of course it's a mix of everything.
Mm-hmm. Thank you, Dr. Kinney. And I do have one question regarding mitigation strategies, for example, for areas like Puerto Rico, where there's not a clear division between the outdoor and the indoor. What strategies do you recommend in terms maybe of engineering recommendations from your side?
Well, in some ways, in some ways you can benefit from the outdoor as indoor, right? Because a lot of times when you close up spaces, you tend to build up pollutant concentration, certainly to higher levels necessarily than you would have in the outdoor environment. Unfortunately, it doesn't allow for control then of, if you have outdoor fungal communities, as you know in our study, you're going to see those penetrate into the indoor spaces. So then what you're trying to do is just so you don't have an overgrowth, an undesirable overgrowth. So really it's about moisture control and, cleaning surfaces and you don't need anything particularly elaborate. Cleaning surfaces, mopping surfaces, those sort of things actually do reduce your exposure level. So some of the simple things I said, like your mom told you to do, some of that actually we should be doing. I think there was the grandma hypothesis. There's also the mom hypothesis that's also proving to be correct.
Thank you so much, Dr. Kinney. Thank you for your presentation, and thank you all for being here attending. Just a few other reminders. To access the next session, click on the open door icon at the bottom left of the screen of the current session, and then hit on preview. And if the session has not yet started, you will see a timer and hear background music until the moderator and speaker or production are in the session. And as Barbara mentions in the comments, please do complete the evaluation of the different sessions. Thank you all, and thank you again, Dr. Kinney.
Thank you.
Hi, everyone, and welcome to the fourth session of the mini-symposium, Microbiomes from the Environment to Human Diseases. We thank you for joining us today. My name is Natalie Melendez-Vasquez, PhD student from the Microbiology and Medical Zoology Department of the UPR Medical Sciences campus, working under Dr. Filipa Godoy, and I'll be your moderator for today's session. Prior to starting our session, I would like to discuss some technical instructions. All participants who are in the session will be able to listen and view the speaker and moderator, as well as the presentation of the speaker. All microphones and webcams from the attendees of the session are not visible. During the question and answer session, attendees interested in asking questions and appearing next to the speaker of the session would need to press grab the mic at the bottom left of the screen. The production will grant access on first come, first served basis. Grab the mic won't be granted by production while the speaker is giving his talk. During the Q&A session, questions can also be written in the chat, and the 5-minute for Q&A will apply to both questions asked live and questions written in the chat. Once the session is over, and for attendees to access the following session, they would need to click on Open the Door icon at the bottom left of the screen of the current session. Once you see the agenda, click on the preview icon that appears to the right of the session title to access the session. If the session has not yet started, a timer and a background music will appear until the moderator and speaker or production are in the session. Important disclaimer. The content of the session and all other sessions cannot be reproduced without prior authorization from the organizer, the Department of Microbiology of the UPR Medical Sciences campus, and the production Vistazo a la Ciencia. Now, for today's session, we will be having Dr. Greg Caporasso with his seminar titled Toward Microbiome Multi-Omics with CHIME2. As a student of the UPR Microbiome Lab, it is my honor to introduce Dr. Caporasso. He is a microbiome expert, being popularly known for developing the famous CHIME2 pipeline for analysis, which he will be explaining today. Dr. Caporasso has published over 100 papers on the human microbiome, environmental microbiome, and microbiome bioinformatics. He leads the development, documentation, community building, and support efforts of the popular CHIME microbiome bioinformatics platform, a very widely used tool in microbiome research, which has been cited over 30,000 times since its initial publication in 2009. Dr. Caporasso is also regularly involved in teaching CHIME2, bioinformatics, and scientific computing. He completed 2 bachelor's degrees, one in computer science and the other in biochemistry, both from the University of Colorado at Boulder. In the same institution, he obtained his biochemistry PhD degree and later continued to his postdoctoral position in the field of bioinformatics and human microbiome. Currently, he is an associate professor in the biological sciences and computer science departments at Northern Arizona University and director of the Center for Applied Microbiome Science in the Pathogen and Microbiome Institute of the same university. His most recent projects have been focused on studying relationships between cancer and the gut and oral microbiome by the very compelling work that has been emerging in this area, linking microbiome composition to the efficacy of cancer treatments, carcinogenesis, and cancer prognosis. Without further ado, I leave the floor to Dr. Greg Caporasso.
Well, thanks so much for that very nice introduction, Natalie. And it's my pleasure to speak here today. Thanks so much to the conference organizers for having me. As Natalie mentioned, I'm going to be talking today primarily about the CHIME2 microbiome bioinformatics platform. And my goal with this talk is to give you some ideas about how you can use these tools to improve your own microbiome research, or how you can get involved if you're interested in contributing to CHIME2, if you're developing methods for microbiome bioinformatics. I invite you to get involved in our community. So like Natalie mentioned, I lead the CHIME2 project, but this is really a global collaboration. And that is well illustrated by the author list on the CHIME2 paper, which we published a couple of years ago. This paper had 112 co-authors on it from 77 different institutions in 10 countries. And all of these individuals contributed in some way to the design, the development, the documentation, or the support of CHIME2. And so again, while I lead this is a global community effort. And I thank all of these folks for being involved. It wouldn't be possible to build this without them. Now, I do just want to mention that CHIME2 succeeded CHIME1 as of January of 2018, and CHIME1 is no longer supported at all. You can still access the old code, but we don't provide technical support for it. And really, CHIME2 is better in all ways than CHIME1. And so if you haven't transitioned, I recommend making that transition. And I'll point at some resources along the way that can help you do that. I'll also mention I have a link. I have many links scattered throughout these slides. At the bottom of this slide, I have a link where you can access these. I will also share these slides with the conference organizers so they can pass them on to you. And I'll have this link again in my acknowledgement slide if you don't have time to jot it down now. Okay, so I want to talk about multi-omics in the context of CHIME2. And I want to just sort of build up there with some of the with sort of the way that I view the different approaches that we are currently using. And so depending on the technique that we're using, we are illuminating different regions of the microbial world. And so if you imagine that the microbial world is illustrated in gray in this figure, you kind of get a glimpse of some portion of that if you use culturing-based approaches. And so you can see this is a relatively small approach. These diagrams are not intended to really illustrate scale in any very quantitative way. But when you start throwing things in like 16S ribosomal RNA sequencing, you may illuminate more of the microbial world using that approach. And depending on what primers you're using, you may get either different size, so you're able to view more or less of the microbial world. And an important thing to remember here is that these are going to be overlapping, but not completely overlapping views. And so, for example, if you use V2 primers, excuse me, so primers that are targeting the hypervariable region 2 of the 16S, you're going to see different microbes than if you target the V4 region, the hypervariable region 4 of the 16S. And I just mentioned that because that is very important when you start thinking about integrating the results of different studies. And so it is possible to do that integration, but you have to be aware that there are biases introduced every step of the way when you're using different protocols. Now, we heard a little bit earlier today about shotgun analysis, and the way that I view shotgun analysis in the context of these amplicon studies is it gives us a much broader view of the microbial world. And so we should be less subject to PCR bias, but we're still probably not observing everything. And so, notably, we might be missing things like RNA viruses. We may be missing things which our extraction techniques don't work very well on. And so if we can't get the DNA out, we can't sequence it. And so all of these different approaches that we're taking are really only going to get us a partial view of the microbial world, and they all have their pros and cons. Now, when I think about multi-omics, I tend to think of different layers of data in analyzing a microbiome. And a useful analogy here is to geographic information systems, or GIS systems. And so if you think of a GIS system, your goal would be to build some sort of a model of the real world. And you would have different layers of data there. And so you might have information about land usage. You might have topographic information, elevation profiles. You might have information about where different parcels are of properties, where roads are, where people live, and so on. And depending on what you're trying to do, you are going to need some of these layers, and you might not need some layers. And so, for example, if you are trying to plan a hiking trip, you might need different layers of data than if you are trying to install wastewater infrastructure, for example. Now, when we think about the layers in a microbiome study, that's where multi-omics comes in. And so the layers that we might be working with, rather than streets and elevations, are things like taxonomy, which we can get from marker gene surveys like the 16S surveys, ITS surveys, some types of shotgun surveys. Another layer might be the functional potential of a microbiome, and that might be something that we get from a shotgun metagenomics survey. We also may be interested in functional activity, and so things like metatranscriptomics, metaproteomics, metabolomics are things that we would get from those types of studies. And we heard Dr. Crandall talk this morning about using shotgun analysis and that being a key to getting higher resolution data. And so if you want to understand, say, what pathogens are present in an environment, you are not going to get that most likely with a broad 16S survey. You might be able to get it with a targeted amplicon survey, targeted to that pathogen. But in general, you're probably going to need something like shotgun data and tools like Dr. Crandall was talking about. We also heard from Dr. Landay this morning, who was talking about metabolomics as essential for developing a mechanistic understanding of the microbiome. And so your 16S survey on its own is not going to tell you which microbes are functioning or what functions they're carrying out. In order to get that information, we need to turn to things like metabolomics, metaproteomics, and so on. And so this is really the goal for where we are now going with CHIME 2, is building a microbiome multi-omics platform. And so we aim to support these different types of studies and, very importantly, provide the tools for integrating them together so that we can learn more from the combination of these layers than we could learn from any of them on their own. Now, you might wonder how we're going to do that, and I'm going to come back to that in just a few minutes. But I want to spend a few minutes now talking about CHIME 2 and some of the features that attract users to the system. And so really, you know, the first thing that people are looking for when they come to CHIME 2 is methods. So they've got microbiome data and they need to turn that data into new knowledge. And they come to CHIME 2 because we have kept pace with the latest methods used in the field, and we provide access to many bioinformatics methods and visualizations that can help you with this. And so this, for us with CHIME 2, really started with doing things like obtaining higher resolution taxonomic profiling. For example, like where we started here 5 years ago or so, is moving from OTU clustering to the use of amplicon sequence variants. One thing that we always try to avoid with CHIME, and that was both CHIME 1 and CHIME 2, is reinventing the wheel. And so if somebody is already doing good work on tools, we aim to integrate those tools rather than replace those tools. And so, for example, if you're using CHIME 2 to define amplicon sequence variants, what's really happening under the hood is that you're using the data2 pipeline to do your quality control and define those sequence variants. And again, I'll come back to exactly how that works a little bit later. These methods also include new functionality that we have developed in the platform. And so a very interesting tool that you may or may not be aware of is in a CHIME 2 plugin called Clawback. But the idea here is that using marker gene data like a 16S profile, but in the context of previous marker gene data, we can build taxonomy classifiers that improve taxonomic classification accuracy. And the way that we do that is effectively by integrating information about how likely we are to observe certain organisms in certain environments. And so the taxonomy classifier becomes environment aware. And so this is a very useful approach for getting higher resolution taxonomy assignments. And it's a better approach than some related techniques. So for example, than building environment-specific databases. If you build an environment-specific database, for example, something that contains only oral or only gut microbiota, you don't really have the ability to identify something that hasn't been previously identified in that environment. When you use an environmentally aware classifier, you can observe those things that haven't been previously observed. You just need more evidence. You need better support for that. So very cool technique. We published this a few years ago in Nature Communications, and you can find information about this through the CHIME documentation. And this is something that we are continuously working on, is supporting what people most need to do in the field. And so we were recently obtained funding for a new project that is going to focus on designing new bioinformatics methods and visualizations for assessment of FMT efficacy. And so we've heard several speakers this morning talk about fecal microbiota transplants. In my view, this is one of the most promising techniques for turning all of this new microbiome information that we have into clinically relevant applications. And we are building tools, just getting started building tools for helping researchers assess if the microbiome transferred successfully in an FMT study, what aspects did or did not transfer, and also in comparing FMT treatments to try and relate the features that transferred to clinical outcomes. And so, for example, if you notice some of your FMT recipients respond to treatment while some don't respond to treatment, we will be building tools to get a better understanding of what were common features in the FMTs that were associated with a positive clinical response. We, like I mentioned, are just getting started on this. If you're working on FMT studies and you're interested in these ideas, please do get in touch. We would love to hear about what you think would be most useful in your study, and potentially even collaborate on some applications of these new tools. And all of this in QIIME 2 is in the context of interactive visualizations to support exploratory analyses. And so these are just a couple of examples of the types of interactive visualizations that we're using and building in QIIME 2. For doing spatial analysis of microbiomes, for doing temporal analysis of microbiomes, and so on. So there are other features that initially attract users to the system. One of those is the accessibility of the system. And so we spend a lot of time thinking about our documentation. We spend a lot of time thinking and evaluating our interfaces with users. And we really focus on trying to build accurate, detailed, and interesting documentation. Additionally, QIIME 2 is completely free and open source. So you have a license right to use QIIME in your work. You can even modify it on your own if you want. We'd rather you contribute if there are things that you would like to see change in there. But this includes commercial use. And so you can also, if you're building a business, you can use QIIME 2 for free in that business. Our goal is really to get these tools out there and to improve the state of microbiome research. Lots of documentation. Some of this is focused at the level of theory, such as my book, An Introduction to Applied Bioinformatics, which you can also find for free online. And then some of this is focused on practice of using QIIME 2. And you can find all of that at docs.qiime2.org. And the last thing that I want to mention here is that people are initially attracted to the system because of the large community of microbiome scientists, developers, and bioinformaticians who are involved in QIIME 2. Now, the hub of the QIIME 2 community is the QIIME 2 forum. This is a free tech support forum. So you can go there to ask technical questions about QIIME 2, to get help if you encounter an error while you're using this, or also just to have general discussion about microbiome research, about protocols, and so on. If you're not already a member of the QIIME 2 forum and you're getting started in microbiome research, I would highly encourage you to join. You can just log in with your Google account or GitHub or Twitter. Don't need to create a new account to do this. And one of the things that is very exciting about what's happening on the forum is that this is becoming a place where community members are sharing their own QIIME 2 related resources. A great example of this is what I have highlighted in the bottom middle of this slide. This was content that was contributed by a Chang2 user in China who translated a lot of our documentation into Chinese. And so this was certainly not something I'm capable of doing, but this was a contribution from a community member. And we try and get our community members really involved and part of the team. And so, for example, this individual for this contribution was invited to co-author the Chang2 paper with us. That remains one of the most popular forum posts, or one of the most popular posts on the Chang2 forum. I want to mention that our community interactions are governed by our code of conduct. And we do that to ensure that we have an environment where people feel comfortable working, asking questions, and interacting with others. I would strongly encourage you to go read our code of conduct, which is linked from this slide. And that really outlines our expectations for how you and other community members will engage with one another in a respectful way in this forum and in general in the Chang2 community. We are also very focused on running workshops. And so we teach theory and hands-on practice of Chang2 at these events. We have done hundreds, well over 100 of these over the years now. Sometimes these will be hosted at institutions. Sometimes these will be hosted at central events. So for example, we sometimes do these before microbiome conferences. And of course, we have recently had to transition all of these activities online in response to the pandemic. And we have actually had a very good experience with hosting these events online. So we have done several workshops. We still focus these on hands-on activities. And so we set up a server that students are logging into, and they can interact with one another, with the instructors, and with Chang2 in much the same way as we do in person. Of course, we miss out a little bit on being able to get together in person, but the flip side of that is we have held some of our most diverse workshops in terms of the geographic diversity. We have had our first online workshop. We had people attending from over 50 countries around the world. And so while we certainly lose out a little bit on the in-person interactions, we gain on making these very broadly accessible. And on that point of accessibility, we now have a full Chang2 workshop on our YouTube channel. And so you can find that channel link up here. And this is actually all of the content from one of our 5-day workshops broken down into video segments that are anywhere from about 10 minutes to about an hour long. Median video length is probably about 20 minutes. And so this is something that you can do on your own time to learn Chang2. And so I encourage you to take a look at the YouTube channel and this playlist. Now, this effort is really focused on increasing diversity and inclusion in microbiome research. We want everybody to be able to access those, and that's why we provide this content for free. We're also in my lab very interested in increasing diversity and inclusion in open source scientific software development and computing in general. As Dr. Crandall mentioned earlier, informatics is becoming an essential skill for scientists, and we hope to assist with training individuals in those techniques. I just want to mention a new project that my lab is getting off the ground that is focused on engaging Native American students in scientific computing. And so Native Americans are very underrepresented in STEM fields. And we aim to change that in open source scientific computing with generous support from the Chan Zuckerberg Initiative. And so I'm based at Northern Arizona University, which, as you can imagine, is in Northern Arizona. We have a large Native American population at NAU. And in collaboration with the other PIs on this project, we are getting a new outreach program off the ground. And so that includes Professors Jenny Ingram and Naomi Lee, who are also here at NAU with me. Jocelyn Lee, who is based at Fort Lewis College. We're also working closely with Mansell Nelson, who is the Senior Program Coordinator for the Institute for Tribal Environmental Professionals at NAU, and Jeff Mylander, a PhD student in my lab, who is the founder of the Flagstaff Eco Ranch, which is a nonprofit focused on sustainable small-scale agriculture, which has already trained thousands of students. And so this is a completely new project for us, but we're partnering with teams that have done this type of work for a long time. And so that includes the CARE project led by Drs. Naomi Lee and Jocelyn Lee, Dr. Jenny Ingram's NIH-funded Partnership for Native American Cancer Prevention, which is currently in its 13th year of funding, and the Institute for Tribal Environmental Professionals, which has been involved in environmental education outreach in tribal communities since the 1990s. So NAU and Fort Lewis College, the two sites where we are carrying out this project, both serve large Native American student populations, so they're excellent sites for this project. And just getting a little bit more specific, our goal for this project is to make young students aware of the field of scientific computing and the opportunities that it provides for building a rewarding career while also working on projects that can bring new technologies and opportunities to their tribal communities. And so to achieve this, we are developing a K through 12 outreach and education program. And it's focused on teaching hands-on scientific computing workshops in schools that primarily serve Native American students, using QIIME 2 as an example project. We're in the planning stages for these right now. We hope to start hosting them in early 2022. And we expect that these are going to focus heavily around the use of Raspberry Pi 400 computers, which is what's sitting in the middle of the desk in that picture. Which we will bring to you and leave at the schools. Okay, but getting back to QIIME 2. So I mentioned some of the features of QIIME 2 that initially attract individuals to the community or to the tool. So things like the latest and greatest methods, the community of scientists who are working on and using QIIME 2. But there's also some other cool things that I want to mention, and I think of these as the lower level features. So these are the things that get people really excited about QIIME 2 once they have started using it. And the first is our decentralized provenance tracking system, which automates bioinformatics record keeping and ensures that the work that you do with QIIME 2 is going to be fully reproducible. And so this system, this retrospective data provenance tracking system, is really designed to help you get away from what I call the "what did you do 5 months ago" problem. And so inevitably, when you run a complex analysis, you are going to have to refer back to that and understand what you did at some point down the road. That might be when a reviewer ask some questions about how you carried out a certain step of your analysis. It might be when some reader of your paper asks for your data or asks for information about your bioinformatics protocol. And if you're lucky, maybe traditionally you would have kept a notes document, maybe you've got all your files somewhere, maybe they're even backed up somewhere. But when you go back to this 5 months ago or 5 months later, even if it's your own notes, it can be difficult to decipher what you did. So for example, we all probably have directories that look like this on our computers, where we've got sample metadata as a spreadsheet and sample metadata 1. And how does that differ from sample metadata old and sample metadata with missing controls? and what is this delete me.txt file, and why do I have a backup of that? And, so when you come back to this, even if it's your own work, but especially if it's somebody else's work, your students or your postdocs, it can be very difficult to decipher this. And so what we did when we built QIIME 2 is that we built the record-keeping functionality into the system itself. And I'm going to just show you a really quick demo of that. And so I'm just going to be looking slightly off screen here as I work through this. But here's one of those interactive visualizations that I showed you before. And so you can see that I'm interacting with this. This particular visualization is tracking microbiome composition over the first 2 years of life in about 30 or 40 subjects. Now, in any visualization that you're looking at in QIIME 2, there is provenance information. And so this initially looks very complex, but this is an illustration of all of the analysis steps in QIIME 2 that got you to this point. And so this circle in the bottom of the graph, and I'm just going to increase my screen size here. So this circle at the bottom of the graph represents that current visualization that you're looking at. And the box that is surrounding it describes how that visualization was generated. And so here, for example, this is telling me that, say I ran this analysis in September of 2018. This was done with a plugin called Longitudinal, an action called feature volatility. And so that is the method that I was using in QIIME 2. If I scroll down here, there's all of the parameters that were used when I ran this individual step. And you can see I can also get information about, for example, like what version of QIIME 2 I was using, even can get really detailed and understand what version of all of the underlying libraries and dependencies that are installed on the were installed on the system when I ran this. And so if I sort of work my way up this graph, you can see that every step in my analysis is annotated here. And so, for example, here, I used an action called filter samples. And so this is where I was filtering some samples out of my analysis. And I can see what the parameters were when I did that. And so you can see, you can get a very detailed and very specific description of what you did. And this tracks all the way back up to the top here, where what I am doing is I am importing some FASTQ files into the system for use in QIIME 2. In this case, you can see I had something like 7 different sequencing runs. And so I've got all of these different import steps up here. But the point is, all of this information is stored behind the scenes by QIIME 2. And so you don't need to record this. It's just there when you need it. So this has proven to be extremely helpful for ensuring reproducibility. And it is also extremely helpful for us, the developers, to provide technical support to users. So we can get a very clear idea of what somebody did and why that might be leading to say an unusual or surprising result. So let me just switch back here. Okay. And so that is how QIIME 2 helps ensure that your work is going to be reproducible. Another one of these lower level features is that there are multiple user interfaces for QIIME 2. And so the same functionality is accessible through a graphical user interface, a command line interface, a Python API. And so what that allows you to do is it allows you to choose the interface that you are going to be most effective working with. And so, for example, users who are more on the biologist, domain scientist end of the spectrum can access QIIME 2 through Galaxy. And so all of QIIME 2's functionality is accessible through a Galaxy interface. And so you don't have to use a command line at all. You can drag and drop and build workflows. If you are more like what I would call like on the power user end of the spectrum, and so you are comfortable using the command line, you are very effective when you log in by SSH to your university's supercomputer or Amazon Web Services, then you can use the Chang2 command line interface. And if you are on the data scientist end of the spectrum, and so you like to work in interfaces like a Python 3 session or a Jupyter Notebook, there is a Python 3 API that you can use. Now, again, what's most important here is that you have access to whichever of these you want to use for any step of an analysis, and all of the same functionality is available through all of these interfaces. And so you don't get, more features if you're a command line user than if you're a Galaxy user. And so what our hope is that this levels the playing field. This makes it so that biologists have an easier time accessing these tools. Now, that Galaxy interface is kind of in an early release. We do have some video on how to use it on the YouTube channel. And we are going to be teaching our first workshop using Galaxy in January, and that is in partnership with the NIH. And so what you can expect, you can either attend that workshop, it may be sold out at this point, but we will be putting a lot of new video content on the YouTube channel in early 2022 that shows how to use Chang2 through this interface. I also just want to mention this one other interface, which you may have seen in there, which is called Chang2 View. That's the one that I was just using. And what that lets you do is view Chang2 outputs. And so it's kind of a read-only interface. And this really supports it really facilitates sharing data with collaborators who may not have Chang2 installed on their computer. And so that is something. We also have a video on the YouTube channel that shows how to use this, how to share data using Dropbox with that, if you're interested in doing that. So very cool interface. I use it probably every day. And so I would encourage you to check that one out. Now, the last thing that I want to mention here is that Chang2 is built on a plugin architecture. And I said earlier, I would give you an idea of how we plan to support all of this different multi-omics functionality. And the key is we are not going to do that in my lab. We are working with third-party developers who are building plugins for their tools to make their tools accessible through Chang2. And so basically what this means is we have an open development platform. And third-party developers can create and distribute their own plugins. What that means for you as a user is that you get access to the latest and greatest functionality without it having to go through my team as a bottleneck. So we are not a bottleneck between the methods developers and the users. What this means for you as a developer, so if you are building your own microbiome methods, is that you can build a Chang2 plugin. You can build information to that, for example, on how your tool should be cited so that you are getting appropriate attribution for your work. But you can distribute that on the Chang2 library. And so what that does is it gets your tool in front of the Chang2 user community. And so it's a way for you to tap into this large community to help promote your software. Just to start wrapping up here, I just want to talk about where we're going, what's next for Chang2. So as I mentioned, Chang2 is going to help you analyze and integrate your microbiome multi-omics data. There are also new ways to use Chang2 on the horizon. I mentioned Galaxy already. We are also working on support for common workflow language. We are going to be integrating Chain2 in new cloud-based platforms that will allow you to access Chain2 on systems where you can rent computational resources. And for all of the folks out there who like to work in R, you will be excited to know that we are building an R API for Chain2. This is currently funded, though we haven't actually started the development effort on this yet. But what this means is that just like you can use a Python API to access all of the Chain2 functionality, you will be able to use an R API to access all of the Chain2 functionality. In the meantime, there's a very cool third-party tool out there called Chain2R that allows you to load Chain2 data in R to work with it using other tools such as Phyloseq. Now, this is something that I am incredibly excited about, and a graduate student named Chris Keefe in my lab is working on this functionality as we speak. And this is something we call provenance replay. And so I just showed you Chain2's data provenance and how it records all of this information. Well, the information that it records is so detailed that we can actually generate new executable code from the data provenance. And so what that means is that probably around the spring of 2022, you will be, or sorry, end of spring of 2022, you will be able to take a Chain2 output file that either you generated or somebody else generated, and using a tool that we develop, you can generate new code from that so that you can either reapply that analysis to the same data or to new data. And so this is another bit of functionality that helps ensure reproducibility and accessibility of these methods. So if you develop a cool way of analyzing data in Chain2, you can sort of build your own pipeline and then export a script that will allow you to do that very quickly. Real quick, we are also working on bringing some of these other features, these low-level features, to other domains, and that includes pathogen tracking. Of course, everybody is probably doing a little bit of SARS-CoV-2 work these days. We have built a tool called Genome Sampler that we used to assist in some tracking that we were doing of the SARS-CoV-2 outbreak in Arizona, where I'm based. And so this is built as a Chain2 plugin so that we have access to this functionality through multiple interfaces. We have all of the provenance tracking that we need. And so I won't spend a lot of time on this right now, but basically this tool supports sampling large collections of genomes, for example, the genomes in GIS-AID, across time, across space, and across viral diversity, so that you can build useful phylogenetic trees, removing sampling bias and applying those for analysis of, say, locally derived data. And so we use this and we built this to help us understand the SARS-CoV-2 outbreak in Arizona. And there's a lot more coming. I have a blog post, which is linked from the bottom right of this slide, where I talk about some of the other things that we are getting started with working on right now. So I would like to wrap up there. Of course, all of this work that I described here today is the result of collaborative efforts. I've got an excellent team working with me, working for me in my lab at NAU. That includes 4 full-time professional software engineers who are working on the Chain2 platform, and then a number of graduate and undergraduate students who are working on different components of these projects. Chain2 is funded by the National Cancer Institute, It has been funded in the past by the National Science Foundation, and my lab is also supported by the Chan Zuckerberg Initiative and the Alfred P. Sloan Foundation. Over here on the right, I have some resources that can help you in learning QIIME 2. And so the documentation is linked in there. You can find the YouTube channel in there. You can find the forum in there. And at the very bottom are the slides that I presented today. And so if there's a link that I had somewhere in here that you would like, I'll leave this slide up. You can just go ahead and type that in and pull these slides up. With that, I would be happy to take any questions you might have.
Thank you so much, Dr. Caporasso, for that very excellent presentation. We will now begin our Q&A session. Right now, we have a question in the chat by Marco Carden, and it says, considering database for taxonomic inference, do you think that QIIME 2 could include NCBI RefSeq database?
Yeah, so QIIME 2, the way I would describe it is it's really reference database agnostic. And so there's no built-in reference databases in QIIME 2. And so people often think of like I often hear people say things like, well, QIIME 2 uses green genes for taxonomy. It's really like most commonly that's what our users are accessing. But you can import any database you want for doing taxonomy assignment. And so there's some relatively straightforward, well, it depends on the data source, I guess, and your skills as a programmer or data scientist. But you can, if you're interested in using the GenBank database, you can format that in such a way that you can use that for taxonomic classification with QIIME 2. Now, if you're working on marker gene studies like 16S, there are databases that are probably better in terms of the taxonomic annotations. And so that is things like Green Genes or Silva, or most recently, the one that I'm most excited about is the Genome Taxonomy database. And so these resources all use NCBI taxonomy as their source, but a lot of effort has gone into the annotation for these specific types of studies, integrating newer taxonomic identities or classifications in them. And so I would recommend typically working with the Genome Taxonomy database is probably where I would get started these days, but you can import other data sources such as NCBI for use in your annotation.
Okay, thank you so much. Production tells me if you could stop sharing the screen, that'd be very helpful. I have a question also. At the beginning, you mentioned the integration of other multi-omics to QIIME 2 to be able to analyze. Do you have any expected release date for that, like integration on QIIME?
Yeah, so there already is some functionality. You can find this in the QIIME 2 library. There is some functionality for doing metabolomics analysis with QIIME 2. There is some limited shotgun metagenomics and metatranscriptomics functionality accessible through QIIME 2, available in the QIIME 2 library. Our focus, like over the next 6 to 8 months, and this is largely being driven by the Bakulich lab at ETH Zurich, is on building shotgun metagenomics workflows for QIIME 2. And so what we expect to have by the end of 2022 is really like an end-to-end pipeline for doing shotgun metagenomics. This is going to include things like read assembly, annotation, all of the things that you would expect to be able to do. a lot of the pieces are coming together now, but we expect to have like fully documented tutorials, videos, things like that. Ideally, we'll even be teaching this in our workshops later in 2022. So there's little bits and pieces coming together right now, and we are starting to like pull those together. And then the real big step, I'm very documentation focused. And so that's like when it really becomes real to me is when there's documented tutorials online on how you do this. And I think that's how it gets in the hands of more users. And so it's one thing, for the experts to be able to use it in these early phases. But when we document it and we teach it and we put content on the YouTube channel, that's when I think of like we really are releasing it into the wild at that point.
Yeah, I understand. Thank you so much for your presentation. I would like to thank you once again for being with us here today. We will have now like our lunch break. I wish everyone to have a nice meal, and we'll see you all back at 2 p.m. for a student career development session. But before heading out, I'll leave you all with Dr. Filipa Godoy.
Thank you, Natalie, and thank you, Greg, for such an amazing talk. And
My pleasure.
I'll have two things that I'd like to ask you. One, I think I've asked you this, especially when we were with Keith in the NCI lab. When can we bring Chime 2 workshop in person to Puerto Rico? Let us know. We are very interested in doing that. We could host you guys here for a week, and you will also enjoy our great weather and students, and
that would be wonderful. And yeah, we would love to do that. And, I really look forward to us, being able to travel more freely again.
Yeah, I think we should definitely talk about this more in detail, because we can have funds, and there are RISE people here who could also help us support the trips and all this. We could definitely talk about it. So one thing that I think it's a very big message that you put out there regarding the data analysis methods is that not only people who are computer scientists can analyze data and can work with Chime 2. So you have a lot of different interfaces that are as user-friendly and as important, like the new Galaxy platform that I'm really looking forward to. And the fact that you are a computer scientist doesn't really give you more tools to work with from the terminal, right? Compared to what you have in the Galaxy platform.
Yep.
So that's good, because we see students that are mainly biologists and have very strong educational background in biology and microbial ecology, but then they lack a lot of the tools to be able to troubleshoot, and they spend a lot of time focusing on the technical details and they forget about the question. So that's really important, In my time, we didn't have the tutorials and we didn't have Chime 2, and we had to go. I remember I went with Ruth Lay to learn ARB in Germany, because we took weeks to get a phylogenetic tree going. So this is a great time to do microbial ecology. And I think this is one of the biggest messages that you left today, is that you don't need to be a computer scientist to work with these amazing tools.
Yep. Yeah, and that's definitely true. And that's a big focus of our team, is making these methods accessible through those other types of interfaces, so that you're not reliant on command line skills, for example, or programming skills. And with the plugin architecture, then the really like third-party developers don't need to worry about building those interfaces. And so if somebody builds a plugin for Chime 2, they, for free, get their tool in all of those different interface types. And so that's how we're really hoping to like make this all more accessible. That said, I do want to just say, like, to your students that, these skills, things like working on the command line, working with Python, they're skills just like any other. If you spend some time on it, a little bit of time every day, and you have a focused plan for how to do it, you can build those skills. And if you build those skills, you will really empower yourself to do modern biological research, which is so data intensive. And so I would, while these tools are getting better, I would still encourage biology students, graduate students, people interested in going into research to work on building some of those skills. Go to a software carpentry workshop, for example. They're only going to help you move more quickly and really like automate away some of the things that are really tedious about working with large data sets. And so they are skills like anything else, like learning to play the piano or speak a foreign language. If you put a little bit of time into it every day and you've got a plan, you can learn how to do this.
Yeah, tremendous advice. Thank you so much. Just to wrap up the morning session, we had amazing talks from rhinos and children's respiratory health to HIV and aging, to the built microbiome and the impacts of living in certain types of homes that it has on human health, and now the broad use of really advanced tools that are free, which is exciting and amazing to see science be so clean and so open and so white to people. It's amazing. It's so reproducible. I think it's one of the most reproducible fields of science today. You can share all these steps of readmes or tutorials that you ended up with these specific figures. You can share that with reviewers, and you can give it as supplementary information in papers. So it's really open science. It's very, very nice, and it's very exciting. Thank you so much, Greg. The afternoon session is for mostly for students, but it's open to anyone who wants to pitch in and give their feedback for students on career development moves and how to build a better portfolio. It's going to be at 2 p.m., so have a nice lunch. Thank you, Natalie, for moderating the session, and thank you, everyone, in the morning sessions and for being here and supporting our Microbiome Symposium. Thank you.
Thank you.